Please take your seats. We're looking at the brink of a quantum materials revolution.
It's about what science can do and what society can become.
Falling walls. What a great image.
Overall, I think it's a huge message of hope.
Here you can do things in 5 minutes that otherwise would take months because you have everybody here, and this is why I'm back every year.
If you do science, you discover that the world is wonderful, and you learn to discover these beautiful things.
It's really one of the foremost science events, I think, globally.
I really enjoy putting my ideas into what's possible in terms of innovation.
Please welcome Sebastian Turner, founder of the Falling Walls Foundation.
Welcome. Luminaries, laureates, Latvians, excellencies, ethnologists, Ecuadorians, members of parliaments and academies, chancellors of universities and states, distinguished guests, dear speakers. Welcome to the 16th Falling Walls Science Summit. It's the 4th day this year, and it may be time that we explain what we are doing with you. This is a collider of unexpected encounters and experiences. Our core experimental device is the coffee waiting line. We managed, whenever you are in a queue, you have two outstanding contacts to get to know, to discuss, and to make new friends. When you see a short coffee line, leave it. Make sure to get to a long coffee line so you have a better experience. Let me introduce just a very few people you may run into if you line up long enough. So, I just received yesterday this wonderful bag from the Falling Walls lab in Erbil. Where is the Erbil crowd? Please raise your hand. There you are, up there. Erbil is not a village in Lower Saxony, as you may think. No, it's in the Kurdish part of Iraq. Another group you will love to encounter is our delegation from Israel. It's the largest group from Israel we ever had, and we are very happy and honored that you are with us. Of course, as in past years, we would love to have all people, all groups from Palestine with us. And whenever I talked to one of our guests from Israel, they said, we will support you. We want this to happen. Thank you. Besides peace, one of our driving big questions we have is climate change. And there's only one excuse you may have not to be here, which is to be at COP30 in Brazil. Some of our distinguished speakers of the past days have already left. Johan Rockström, for instance, who had an incredible presentation 2 days ago, went right away to the airport. But a couple of people from Brazil are with us. Among them, the winner from the Falling Walls Lab. Where are you? Don't be shy. We won't tell that you are not in Brazil now. I can't see the hand, but I'm sure you are here. Where are you? There. Wonderful. Good to have you. Another person decided, if you are here, I can afford to be in Brazil. It's the Chancellor of the Federal Republic of Germany, and he has an address for you that you will see now.
Esteemed guests and organizers from the Falling Walls Foundation, esteemed researchers from all around the world, it gives me a great pleasure to welcome you to Berlin. There could be no more meaningful way to mark this day, the 9th of November, than by bringing together brilliant minds from all around the world to present and discuss solutions to the most pressing challenges of our time. The 9th of November is a pivotal day in German history, one that marks decisive turning points. We not only celebrate the anniversary of the peaceful fall of the Berlin Wall on this day, we also remember the 9th of November 1918, when the first German Republic was proclaimed and the monarchy came to an end. But, most painfully, the 9th of November also marks the anniversary of the Nazi pogroms in 1938, reminding us of the countless victims of that dark chapter in our history. I would like to express our profound gratitude and deep respect to the many guests from Israel who have accepted our invitation to Germany. Your presence here is a great honor for all of us. I would have loved to be with you today, but I have at least been told that you prefer me to be in Belém, Brazil, at COP30. In order to make our world a better place and to preserve this planet, we need both the dedication of researchers and the power of multilateral cooperation. This is why I choose Belém over Berlin today, to join many of your colleagues and many of mine in addressing climate change through collaborative action. In our times, both multilateralism and independent research are increasingly under pressure. The German government is committed to supporting independent research and strong academic institutions. We don't want science to make it easy for us. We want you to do excellent work for humanity. We deeply appreciate your pursuit of knowledge and your efforts in breaking down the walls that still divide us today. I wish you an inspiring day in Berlin today. I hope to see you here again on many more occasions, perhaps even joining a German research institution one day. Thank you and enjoy the summit in Berlin. Most welcome.
With the Chancellor away, well, first of all, when have you ever recently heard a head of government saying to scientists, don't be easy to us? Just be excellent. If the Chancellor is abroad, it offers opportunities for us, because the person who will now address us in person is the individual who has to sign the annual support for this conference. It's the State Secretary, Mr. Plyer, of the German Ministry, the Federal Ministry for Research, Technology, and Space. And I learned that you grew up with 3 sisters, and that made you an even better manager. I believe that one, since I have 3 daughters and 1 son. When you are now approaching us, each and every of you can give an applause for past support, which may be an indication for the appreciation of future support. State Secretary, the floor is yours.
Mr. Turner, Professor Mlynek, ladies and gentlemen, a privilege to speak to you today on this Breakthrough Day here in Berlin, and it does not happen every day that I have the honor to speak directly after my Chancellor. And it's a special pleasure to speak and to be with you here in Berlin on this very distinct day for Germany, reminding us of glory and of dark parts of our history. And I must say I'm thrilled to see so many people here on a Sunday morning at 9 o'clock. So this means you choose this event over a nice breakfast in your hotel or over a nice breakfast with your family. And some of you may have just returned from a club, so you preferred even being longer in the club. And I think this shows that this is an important event for all of us. And I think, if I remember, 36 years ago, we all spent the nights at our TVs looking at the wall coming down. And on the next day, very early in the morning, maybe even before 9 o'clock, I left what I did and I traveled to Berlin, which was for me 600 kilometers, to be part of this. To be part of this very historical moment, which I'm always remembered when I walk through this city and cross the line between the former East and West every day. I think a Sunday morning is also a perfect time to pause and reflect. The name Falling Walls reminds us that walls that once seemed unbreachable have fallen throughout history. The fall of the Berlin Wall reshaped the world. You, the distinguished researchers here today, are also working to overcome walls through your scientific endeavors. Today's event is not just an opportunity to discuss your ideas, but to celebrate your breakthroughs. Every year, the Falling Walls Breakthrough Day reminds us that borders and boundaries can fall peacefully thanks to scientific curiosity, knowledge, and cooperation, and above all, because of your hard work. Scientific breakthroughs are the engine that drives our future. When we talk about quantum technologies, artificial intelligence, biotechnology, or climate-neutral energies, we are not talking about abstract research topics, but about concrete answers to the pressing challenges of our time: climate change, human health, sustainable mobility, and the security of our societies. Research is an end in itself, but it also serves society and creates knowledge that provides guidance and opens up new futures. Research inspires because it shows what becomes possible when we tear down walls between disciplines as well as geographical ones, but also walls in our thinking. A breakthrough rarely happens in isolation. It happens where ideas collide, where different perspectives come together, where people have the courage to think outside the box and ask and explore entirely new questions. That is exactly what we are celebrating today on the Falling Walls Breakthrough Day 2025. Today's program reflects how broadly this breaking down of walls can be understood. For example, we will learn how basic biomedical research is opening up new ways to better understand human fertility, aging and health, for example, in the projects of Professor Melanie Schuh. We will hear from Catalin Carico about how mRNA innovations can overcome genetic barriers. This year's Falling Walls Women's Impact Award winner, Professor Angela Gonçalves, exemplifies a new generation of female researchers who are breaking down barriers between disciplines, technologies, and societal issues. Her work combines molecular biology, artificial intelligence, and clinical research to detect diseases earlier and advance gender equality in medicine. Berlin-based artist Marco Barotti combines art and science, using solar-powered sculptures that reproduce sounds from reefs to help damaged corals regenerate. Dr. Miriam Denhoff shows how social science research can improve the lives of children and families in trouble-prone regions. So maybe I have spoiled you enough, but what I want to say is we need to talk about these breakthroughs and the opportunities that they provide a lot more. Among policymakers in academia and society, successful science communication is crucial for this. And that is one of the reasons why our ministry is funding the Breakthrough Day, because we want to translate research, make it understandable, and thus achieve the broad acceptance that is necessary for a successful transfer of research and innovation into people's everyday life. Science communication is a cross-cutting task for all the stakeholders. That is why we want to take science communication to a new level. This can only be achieved through a national alliance of all stakeholders. We want to give science a strong voice in Europe and the world, and we are starting in Germany. As part of the next Science Year on Medicine of the Future, we want to make science communication tangible for everyone through interactive, participatory formats. After all, science needs broad acceptance. It needs a society that understands, supports, and propels it forward. Innovation, which emerges from research, is essential for our future. That's why the German government has realigned its research policy. The High-Tech Agenda Germany is the government's promise to commit to high-impact, future-oriented, cutting-edge research. The federal government alone will invest 18 billion euros in this endeavor during this legislative period. But be assured, this event does not cost 80 billion euros. The focus is on 6 key technologies: artificial intelligence, quantum technologies, microelectronics, biotechnology, fusion research, and climate-neutral energy production and mobility. The agenda encompasses not only basic research, but the entire innovation and value creation process, from idea to transfer to application. The High-Tech Agenda Germany is about competitiveness and sovereignty, about breathing new life into Made in Germany, trademark by strengthening Germany as a place for research and innovation. Key to this is also the recruitment of skilled workers, international talent, and the best minds in research, but also cooperation to promote a modern innovation infrastructure. For us, the High-Tech Agenda Germany is therefore also a move at international level. We have the opportunity to shape the future through joint effort in Germany and Europe and the world. Because no country can shine on its own today. Research and innovation are global. International cooperation is not a luxury, but rather a necessity, a prerequisite for the breakthroughs we are talking about today. Germany's high-tech agenda explicitly includes the international dimension. And that's what we are emphasizing here today. Science breaks down walls. Even and especially at a time when new walls are being put up in many places. Cooperation in science and innovation builds bridges between cultures, transcending narrow national interests. We live in a time when technological upheavals, geopolitical tensions, economic competition, and new societal problems are challenging us more than ever before. So your collaboration as researchers, as entrepreneurs, as industry partners, as international colleagues in politics is crucial to overcoming these challenges. Your creativity, your transfer of knowledge, your openness to cooperation and to thinking beyond boundaries makes all the difference. Germany, Europe, and the world need not only good ideas, but also courageous action. New business models, new partnerships, new ways of working together. And we need people who seize opportunities and think beyond boundaries. As we gather today on Falling Walls Breakthrough Day, we honor the results of your ideas. And at the same time, we take a look ahead. The high-tech agenda is our starting point in Germany. So let us work together to create a new spirit of optimism in Germany, in Europe, and around the world. Let us break down walls together so that research benefits us all. I would like to thank the organizers, all participants and partners, and wish us all an inspiring day today with lots of ideas, exciting encounters, and clear steps towards the future. Let us break through. Thank you very much.
Yeah. Can't you become the President of the United States of America?
No, because I was not born in the United States.
He says he was not born in the United States. That's the part of the Constitution they should change. I already introduced you to the principles or the core effect, making the weighting lines, new friends colliders. But I would like to share two more insights. Falling Walls tries as well to break the walls between science and the public, the people who fund this by their taxes. And of course, in this room, in this building, over the last days, mostly the people were science enthusiasts, working in science, being related to science. But we feel our mission cannot be to bring people in who say, I don't speak English, and it's so difficult, and it may even turn them off, but rather to identify where is the bridging point between what scientists do and what people love, what children love, what everyone loves. And we learned this in a collaboration with the Bosch Foundation. What really unites everyone in science and almost everyone not in science is the love for puzzling. The joy of puzzling. Research shows that you guys, you scientists, are not so much interested in the results of your work, but in the way to find them, because it's so puzzling and challenging. This inspired us to make a series of photographies that you may see in this very second. You've seen them downstairs, you've seen them on a poster wall outside. There they are. And now let me tell you, we want all of you to have such a picture taken. The photographer, Dominic, where are you?
Downstairs taking pictures.
No, he is in the room. Where are you? Raise your hand. There he is. Can you see him? He's looking like that. So Dominic is located with his little studio on the other side of the photo wall. So from the entrance right away through. Before you kick everything I stored here out, please let me take first this. So we would like to have a picture of you, and if there may be a line, enjoy it. Because our speakers, the scientists, the best of the best who will present their work today, are so thrilled, are so enthusiastic of their work. I'm looking at you, and I'm totally convinced that I'm right in your case, as in so many. It is an incredible task that we asked them for, to be in time. Each and every speaker has only 15 minutes. And imagine the issue you devote your life to should be presented within 15 minutes. That is just outrageous. So we provide a little help. The first help is here in front of me. It's a countdown clock. It tells me that I'm over time. And what happens when you are close to overtime, you listen to this. If you hear this, you are short of meeting another new friend. If you are not, then ending with a few seconds. And that is Klaus. Klaus is with us for many years. Klaus can do many things like this, and he will take away any attention from your presentation. So it's a pretty brutal way, but I mean, look at him. He is big, and so he can have. If he doesn't take away the attention, he takes away you. So please try to come to an end. The program starts now, and each and every to remember the days in Berlin will receive a couple of toys to puzzle further. Thank you. Enjoy the day.
Breaking the wall of RNA therapeutics. How modified RNA drugs are reaching previously inaccessible parts of the body. Anastasia Khvorova, UMass Chan Medical School.
On November the 9th, 1989, I was in Moscow, watching with admiration and hope.
It's a great pleasure to be here, and what I will tell you today is a story of beauty in science, as well as breaking multiple walls to create a path towards transformative therapeutics for Huntington disease, as well as any other neurodegenerative diseases, and eventually aging of the brain. So this molecule is gorgeous. And it represents the way our cell control transfer of information. So life is information. And inside the nucleus, the information is being stored in double-stranded nucleic acid called DNA. And what you can see, it's a consequence of the letters. Yeah, blue, red, yellow, magenta, which is A, T, G, C. And every single one of them, your genomes are different, but they all consist from these different letters, which is information. Information is stored, but then it's need to be transferred, and it's being copied in a single-stranded molecule called mRNA, and Catherine will talk about making drugs out of those long mRNAs. It gets in a cytoplasm, and then it's need to be translated into function. So it's translated into proteins, which is performing vast majority of functions inside the cell. Whenever such a complex informational process is happening, there is, of course, a lot of mistakes can happen. And cells develop a normal, natural mechanism. You're spending 5% of energy doing that, I'll call RNA interference or RNase-induced silencing complex. In this case, it can be programmed with small pieces of this information, like Google, which finds the targets and eliminates it from being able to translate it into the function. So practically, life is a storage of information, information transfer, and information interpretation into function. And every single disease, so 99% of diseases, it's either too much protein, not enough protein, or a wrong protein being made. And RNA interference mechanism, by blocking information transfer, provides a potential for a universal drug. So the human genome is sequenced, is known. It took us a while. It took over, the final version is available in 2022. But because the sequence of the genome is known, it's created an opportunity to actually utilize this natural mechanism to silence any gene on demand. I'm representing here RNA Therapeutic Institute, UMass Chan, and this is a premier institution which is contributing to discovery, understanding fundamental mechanism, as well as translating it into the drug, which is evidenced by Victor Ambros and Craig Miller actually receiving Nobel Prize for discovery of this fundamental mechanism, and Phil Zymer is the one who demonstrated that it can work in human cells. And the story you need to remember is that, yes, it's potentially a universal drug. As with any technology, whether it's a fusion, whether it's AI, whether it's such a fundamental class of drugs, it went through the normal hype curve. What the hype curve means that something cool is discovered, everybody jumps on, things are moved into clinic, like big pharma is pouring money, and then nothing works. And when nothing works, what's happening? Everybody exits. Yeah, it's a very, you can see it with antibodies, with almost any break-in-the-wall technology. And when everybody exits, chemists, they come. And as a good friend of mine, Mana Marharan, who invented most of the chemistry platform in this field, saying, stay, if you don't know what to do, stay calm and do more chemistry. And indeed, this is what happened. And over the decade, the molecules get evolved. The reason why they failed initially, because the half-life of the fully natural siRNA in the blood of the starving student is around 5 seconds. And it's a negatively charged stick which doesn't go anywhere. So with dressing it in a complex chemical modification, we got it to the point that it's actually become a drug. And right now there's 7 approved drugs and many dozen in the final stages of clinical development. And all of that is because of the fundamental complex chemical innovation. So what is today in clinic is compounds are fully chemically modified. They're not RNA anymore. There's not a single RNA base left. It's a complex pattern, and then its structure, where it's conjugated to the gallneck, which goes to the liver, or lipophilic entity, which broadly distribute, or antibody, or more complex architecture, that is what driving the delivery. The reason why full chemical stabilization is so important is that when those drugs get inside the cells, they're entrapped in cellular compartments called lysosomes, which is very, very aggressive for degradation of proteins. And as you see over here, it's like red siRNA entrapped in a green endosome. And when it's stabilized, those endosomes serve as our big friends because they provided an intracellular depot of the drug, which has been slowly released over time. And this is why in approved therapeutic today, think about it, how long do you think the clinical efficacy of the single administration? First time when I saw the data, it's really breaking the wall because it's up to a year. Think about it. It's a potential to change a daily pill compliance, doctor's visits, prescription, pharmacy administration, to semi-vaccination to prevent your high cholesterol, where inclisiran, it's an existing approved drug today, which probably everybody over 50 should be taking, and vaccination against Alzheimer, making sure that you have tons of muscle, etc. Of course, all approved drugs right now is in the liver, and I got dragged into the field of Huntington by a good friend of mine, Neil Aroning, and he said, okay, Anastasia, you need to deliver those things to the brain. And at the time, it was not only impossible task, it's a completely crazy thing. And Huntington is a devastating disorder which is caused by expansion of the CAG repeat in the first exon of the gene, and it's causing all those devastating consequences named here. But in a late age, people are born normal, they go to graduate school, they become famous politicians, journalists, and then at 40 or 80 age, it's a severe cognitive neuropsychiatric disorder with a very quick decline. So to be able to develop a drug for Huntington, you need to deliver it to the brain. And the three very talented students and postdocs working in my lab, they accidentally discover that if instead of one molecule, you link them together, you make them larger, you get a profound enhancement in the retention in the brain. And we got what is called pink brains. It's a brains which is turning pink, which is indicative of the siRNA drug being inside the brain. We got it in a large brain with essentially delivery to every single neuron. Okay, getting drug in the brain is a good thing, but you need to eliminate this protein, which is causing function, and we were able to show that the Huntington protein is gone. Exciting. Yeah, I gave a talk in the Huntington Patient Disease Forum, and we got a standing ovation because there is finally a hope for a treatment. We start testing this compound in animal model of disease, and nothing happens. And that was super disappointing. Before there is another breakthrough, falling wall. It's a discovery by many groups indicating that in Huntington disease, the RNA and DNA, this information transfer, it's actually contributing to disease as much as a protein. And later on, we and others have figured out that when you have those expanded repeats, so it's repeated words, it's when information is being transferred, it's kind of misfold itself in the wrong way. And because it's misfold itself in the wrong way, natural mechanism of maintaining DNA integrity try to correct it. But when it's trying to correct something, it actually creates more issue. And the issue is expanding of this repeat from 40 to 50, and when it's reached 150, the neuron dies. And this is why it's taken such a long time for disease to manifest. We figure out that if you silence the key target in this pathway called MSH3, you actually can block this expansion. So two processes, somatic repeat expansion and mutant Huntington, it's contributing to the disease progression. So what do you do? There is a very nice model, animal model of the Huntington disease progression, which essentially over the months, several months, you can see this armadillo shape appear, which indicates there is more and more expansion. So there is expansion on the right. It also forms the protein aggregates you can see in patients as well, and most informally, the information is disturbed, like this transcriptional signature is all over the place, correct? So it recaptures the features of the Huntington disease. So what we did next, it did a large study, which took a lot of money and a lot of time, and treated, block for a year in this animal model expression of the disease protein, block somatic repeat expansion, all the controls, and a combination of the above. And what you can see is that if you look at this manifestation of the disease, those protein aggregates, if you remove the protein itself, nothing really is happening. If you block this genetic expansion, it reduces profoundly. And if you do both, the effects are cumulatively and profound, indicating that we might be up to something. But finally, we look at this distortion of the transcriptional signature, and what you see in a gray is the genes which change between disease, between patients and normal people. So there is 161 changes. It's like the wall, yeah, the gray wall. And when you block somatic repeat expansion, like 30% of the wall is gone. And when you do both, you are breaking 50% of the wall, which is potentially disease modifying. And mice, at least, are doing very, very well. So what I wanted to emphasize that when we are talking about the major breakthrough in the science, it's not one thing at a time. For development of the path towards transformative therapeutic for Huntington disease, and you understand it's an informational drug. The same technology right now is being used for treatment of Alzheimer, Parkinson, ALS, many orphan indications, and third of my lab is working on the rejuvenating of the brain. Of course, I don't want to age. But it requires like breakthrough discovery of RNAi, discovery of chemical stabilization, brain delivery, sequencing, Huntington gene, driver, combinatorial silencing, and this is providing a path towards their Huntington cure. And the last thing I wanted to say is that it really takes thousands of scientists in many countries working together on the fundamental basic things to get towards this type of transformative things, and funding basic science is very important, if not in the US, at least in Germany. And thank you for a great team.
Breaking the wall of traditional education. How new approaches are shaping the future of learning. Peter Schlosser, Arizona State University.
On November 9th, 1989, I was in Germany for a conference, watching the news on TV.
Good morning. It's great to be here. As you can see, what we want to talk about today is the future of not just education, but the role of academia as a whole. So let's start with two questions, just by show of hand. Who of you is concerned about the future we are heading in? Easy one. Who of you believes that universities are doing all they can do to help us navigate into the future safely? All right, there are a few, but very, very few. So there is a little bit of optimism, but there's a lot to do. So let's see where we are when we are looking at universities and their role through time. There's, of course, evolution, as there is in any part of the world. Universities started actually as entities who holistically looked at the world we live in and what that means for us in terms of knowledge gathering and applying that knowledge to societal issues. As time went on, there was an evolution from that holistic perspective to one that was more discipline-oriented. And a lot of good came off that. And we saw an example just now. But as we are moving forward into a world that's more and more complex, where issues such as climate change that have been mentioned before, others like public health, like safety of food, societal stability, are highly interlinked, we have to come back to looking holistically at the world we live in, understand how did we get here, why are there so many pressure points, and what can we do in understanding that, and more importantly, envision a world in which the future generations still have choices, rather than dealing with all the problems we have left for them. And that is anchored in system thinking and in a holistic view of where we are heading. But we are actually still moving in academia mainly along a system that was designed for a different era. It was designed for uniformity, repetition, predictability, and actually a slow pace of change for a long period of time, for the industrial period. That doesn't mean to say there was no change. There was always evolution, and there will be. But the question is, are we evolving fast enough? Let's just have another question, just show of hands. Who of you remembers being taught on a black or white board? Who of you that doesn't work? Anyway, this should be an animation. But who of you is actually teaching using immersive technology, including virtual reality? A few, not that many. The difference between teaching on a blackboard and teaching in virtual reality is a little bit like the difference of the world we lived in a few decades ago and the world we are moving in at a rapid pace. If we are looking at the world today, what we are seeing is changes at a very rapid pace. If you look at that upper left figure, you can see an exponential increase called the Great Acceleration, starting at about 1960, the last in a cascade of accelerations that started with the Industrial Revolution. And that pervades all parts of our life. It includes climate change, droughts, floods. It includes societal stability, food security, and the ubiquitous evolution and rapid evolution of what is typically called artificial intelligence, which I typically call accelerated integration. So now, in such a world, what is the role of academia? If we are looking at academia and its evolution, in the US, we often characterize that by waves. First wave was the Greek academies. Then it goes through several, typically in this case modeled after the US system. You have the state colleges, etc. Let's jump to the fourth wave, which are research universities. Then the fifth wave, which are national service universities. They take actually research universities and combine them with scale and accessibility with world-class research enterprises. But even that is not enough. We have to think about the next wave, which is the university of the future. It will be dynamic, a structure that adjusts in real time to a rapidly changing world, and it's characterized by being deeply integrated into society, co-creating, being societally relevant, acting with urgency, having direct impact, working at scale, and being globally connected. So at Arizona State University, we are experimenting with academic structures. The aspiration is to create a so-called new university, and that actually requires new organizational design, and that manifests itself to move from the typical approach to an enterprise approach, and the focus is completely on scale, speed, and impact. That does not mean that basic research is not important. it is the foundation, but it has to be augmented by applied research that translate much more rapidly what we learn in basic research into impact in society. So what do we mean by scale, speed, and impact? Just based on ASU's evolution, here are a few numbers to highlight that. On the scale side, we have close to 200,000 students. About 100,000 of them are taught digitally. 500,000 students get courses from us for credit. We have students from 165 plus countries. And to be really able to experiment, we are pushing philanthropy. Right now we are at about 300 million a year. Speed, we can set up a new school, a new department, a new college in less than 6 months. And impact, I'm not hung up on rankings, but they tell you a little bit of something. So we are ranked #1 in innovation for 11 years in a row since the beginning of the rankings, and #1 in impact since the beginning, 6 years since the beginning of those rankings. So there is a mix-up in the slides. So what we are doing actually is we are creating a new type of university called the Global Futures Laboratory, which in essence is a mini university embedded in a macro university to be incubated until it can stand on its own. The way it is designed is to have, as a university would have, a discovery space or research, a learning space or education, but we added a solution space as one of the fundamental spaces that we have to work in, and that is informed by discovery and learning, but also informs how we learn and how we discover. We need networks. The most important space that we created is probably on the lower left, the engagement space. That is a space where we dedicate a lot of our effort in understanding what society needs, how to communicate with society, how to understand which knowledge they need for the decisions they have to make today. Do we have that knowledge and not communicating it? Can we create it? Or how are we actually co-creating that? The framework of that new university is transforming complex systems through innovation. And we are moving from disciplines to themes. So, and then if you combine the themes, you have the ability to holistically look at our planet. If we are looking at this slide, what that actually highlights is that we have to break the wall between knowing and doing. Often we are stuck too much in knowing and not translating it into doing. So if we are looking at the picture here and start at the lower at the bottom here, we have a growing population that puts more pressure on the Earth system. We have to look for responses to that pressure, and we actually find a lot of them. It's an important point. We have more knowledge than we are applying. If we find these solutions, we have to look if they are economically feasible. If they are, we have to look at whether or not there are institutions to implement them. And a lot of what we are doing passes all these filters. It's all necessary. We are working on all of them, but it's not sufficient. If we are not getting at that inner core, which is decision-making, which is understanding how humans, here shorthanded as societal will, are taking information and applying it, then a lot of the knowledge that we are gathering on the outside is in essence meaningless for the way the world moves forward. Just a few examples of how we are actually working that in actuality. So I talked about these spaces. Let's look at the engagement space. There we have a major effort in advanced decision support for complex issues called the Decision Theater. It has been showcased here twice, last year and this year. So if you have more interest in that, there is a lot of material about that. That brings together stakeholders from around society to contemplate complex issues and play through scenarios in terms of what would be the outcome if you go one path or another path. So that's co-creation. It has different levels, visualization, scenario formation, and we are now moving towards a point where this tool can become a virtual participant in the discussion about the future. Let's look at the solution space. We have created, or we have a project, a large project, that deals with the future of the Southwest. The Southwest of the United States is under several pressures, getting hotter, getting drier, population growth. So we are moving around the economic future under issues that are combining issues that relate to water, energy, climate, carbon, air quality, and workforce. The major issues that determine where the future will be and where the economy will be. We are running that like a private sector enterprise. We have a CEO running it. I was the PI as a scientist, but immediately hired a CEO, a COO, a CIO, a Chief Innovation Officer. So we are running that very differently. Last one is the learning space. And here we are actually using virtual reality to bring students, learners around the world, including some of the spaces that are changing most rapidly, such as the Arctic Ocean. So we bring them into the Arctic. You see a picture here from under the sea ice, and hopefully this one moves forward. That should be an animation, and the animations are not kicking in, but you get a sense here. We take them, the students are pretty much on the ice, they feel the wind, they hear the ice cracking, they are operating instrumentation, such as the remotely operated vehicle. They go with it underwater, they measure the ice, they take the data back into the glass and actually have a fully immersive experience. We take them into coral reefs, into ancient cities, so that they understand for whatever sliver they are responsible for in their job to make decisions, to put them into the context of the entire world. So, and by the way, there is a demonstration of Dreamscape Learn of this virtual reality outside here, starting at 10:30. So, future is now. We have to act. For those of you who are running universities, I would appeal to you, be bold, experiment. For the young generation, take the agency. It's your world. Make sure that you get the space that you should have. And final question. Who is ready to embark on these experiments? Quite a few. That is encouraging. So let's go from contemplating to doing. And I'm looking forward to work with many of you on this kind of transformation of academia. Thank you.
Breaking the wall of carbon-neutral cities. How urban green interventions reduce greenhouse gas emissions. Zara Kalantari, KTH Royal Institute of Technology.
Good morning, everyone. It's really a great pleasure to be here. So what I'm going to talk about is a bit of quick journey through the time. So let's start with this, a planet in balance. Yeah? Imagine a planet that we didn't exist. It was in harmony. And then we came in, humanity. We are curious. We are inventive, ambitious. So we started to learn actually how to reshape our surroundings, how to harness the power of fire, water, and wind. So we started to build our cities. That was a magnificent symbol of culture, innovation, progress. So our city became like a heartbeat of our life. We started to communicate with each other. We started to build with each other. But at the same time, so we started to reshape our surroundings, redirecting rivers, clear-cutting forests, and the balance that was existing in this planet started to tilt by itself. So, as we see, the planet that was in harmony so started to get affected by our changes. And the planet was responding back. How? Bringing, for example, the disasters, flooding, the heat waves, the fire, increasing in CO2. So what we see actually that by changing the surroundings, by changing the environment that we are living in, so we started to see that if we want to keep the sustainability and resilience in our surroundings, we cannot just have the silo solutions. We need to think like, okay, we are created a complex web of interconnected challenges. So these challenges, they are connected to urbanization, climate, human health, emissions, water quality, and so on. And we know that urbanization, we are living in a planet that the age of urbanization started long time back. So at the moment, we are 8.2 million, and we will go to 9.7 million in 2050. And what we can see in this figure is that more than half of this planet's population lives in cities. By 2050, which is not far, far away, actually 2/3 of this planet population is going to live in cities. So through the time, we have seen that actually we can see that our cities have grown. So for example, from Manus, from Amazon rainforest, we have built the cities that actually we cannot see any more forest there. So here is a picture of Rio, from Atlantic forest to today. Another picture, LA. Mexico City, Maldives, New Delhi. So all these cities, they have the same story, yeah? Forests replaced by buildings. Coast turned to harbors. The natural balance traded for our own urban growth. And so, we can say that we as a human being actually reshaped the earth skin by our hands. And we saw it in Johan Rockström's presentation that the planet is speaking back. So we are almost off the charts, literally, yeah? So the global temperature have climbed to the record-breaking levels, as we see. So we began as small changes through the time, and now we have accelerated to a really wild imbalance. So the Earth that once was nurturing us now demands our attention. And the building sector that we are living in, we have our structures, we have the roads, we have transport, that we have actually our home there. They are transmitting. They are one of the major global greenhouse gas sectors. And we are reshaping the face of our planet faster than any other time in history. So what took nature billions of years actually to evolve, we are altering in mere decades. So here are examples of what has happened. The impact and the evidence, we see it, is everywhere. There are many examples of the climate disasters. I have just put some of them from the recent years. The fire in LA in January 2025, the flood in Libya. And actually what we see that they have resulted to economy loss, they have resulted in fatalities, the replacement of the people. And we had recently the storms in Europe, flood in Valencia. So I can say that each event is a chapter in the same global story. That the story of our planet that is going out of balance. So economic losses stored into billions of homes, livelihood, and also destroying and replacing and displacing people. So as we see, and as we saw actually in those cities, in those examples, climate change is not any longer an abstract. So it is lived daily by millions of us. And I can say that by disconnecting strategies, solutions, responses to this climate threat, we have built a wall that we need to break through it. So this is the time to reimagine resilience. This is the time to rethink our cities that can adapt, to recover, to transform, and be a foundation of our shared future. So then the question comes to us as scientists, as planners, decision makers, policy makers, that which solutions do we need really to be able to enhance the resilience of our cities, to do better climate adaptation and mitigation? So this question has been with us for a long, long time. And now we have come to a new path, a new approach that is emerging, inspired by cooperating with nature, not against nature. So these are the solutions that can make cities more livable, more resilient, and more human. So this is an example from New York in 1930. This is the High Line. Maybe some of you have seen it, but this is a place that actually we have put one of the engineering solutions to an urban problem that was fast, functional, and entirely artificial. So what happens through the time? This railway was abandoned and then reborn again as a High Line park, so which is a green corridor, alive with plants, art, and people. So another example is from Copenhagen 8 House, where the green roofs, water retention, the landscape design, they have been built, embedded to the DNA of the city. And this is beautiful, how you can put these things into your DNA, yeah? And we have also another scale of nature-based solutions. For example, in Madrid, 120-hectare green corridors, they have been built to reconnect not only the ecosystem, but us as well, the communities and the ecosystem together. So we can see that cities around the world are beginning to embrace nature-based solutions, but we have still these questions unanswered. How and where we need to apply them? This is very important. Don't take it easy, really. It's very important. So, and that's the reason we started with so many projects. Each of these bubbles are the projects, that we're working together to make science meeting solutions, to answer these questions. And for example, we started a new project that building an AI-powered knowledge integration to carbon-neutral cities. And this project has been recognized by the Frontier Planet Prize, so for its transformative potential to address global environmental challenges. So what we are doing, we are trying to help the cities to unlock the full potential of the nature-based solutions, not to only make it greener, make it healthier environment, but also to achieve the carbon neutrality in our cities. So the question that we have put forward is that is really nature-based solution can truly reduce the carbon emissions. So having this question in mind, we have put 3 other more fundamental questions as well that we are trying to answer it. So one was, which type of nature-based solutions strategies are most effective in reducing carbon emissions? So if you are looking at the nature-based solutions, it's an umbrella of different solutions. So it's not just one solution. But when it comes to the carbon neutrality, to capturing, to reducing, actually to have the direct and indirect impact, so we need to look at which of them they are most effective. The second question is, what are the potential contributions? So we have to do the calculation. We have to do it, do the math, to be able to understand what are the potential of nature-based solutions to achieving carbon neutrality. And the third one is where we need to put them, because this matters a lot. When I'm talking with the city planners, they tell me, where should I put it? I want to build that, but I don't know. Where should I put it to have the most impact here? And this is where we want to talk with you, actually, and saying that, okay, if you want to put it here and there, it's going to have huge impact. So, if we want to transform our cities, are you with me? Yeah, yes? If we want to transform our cities, it matters a lot where we put those solutions. For example, imagine, so we have the colleagues coming here now with the trees. Woo! So imagine, just imagine, okay? Here is the Kungsgatan in Stockholm, yeah? And I'm going to plant trees, but I don't know where I should put it. But I have made a huge calculation there, a map there, yeah? So let's see. This is Kungsgatan. You are in Sweden, in Stockholm. I'm walking away. And I'm just checking. Oh, yes, that area. So can you please come with me? So maybe here, yeah. Do you mind? Okay, so, yeah. What my map says, yes, maybe here. Oh, yeah. Do you mind?
Thank you.
Oh, fantastic. So, please come with me. So we are walking in Stockholm in Kungsgatan. Hi, how are you? Hur mår du? Mår du bra? Yeah? Okay, so, and yeah, maybe I have to put it there. Can you please move a little bit more? Yes, that's going to be nice. Okay, good. And then I have plenty of the trees, but a very long road as well. So please come in here. That's going to be great. Oh, why? My map says there as well. So please come in. Yes, yes. Do you mind? Yeah? So, yeah. No, not there. Yeah, we should move it. Move it a little bit back. Yes, yes. Oh, more trees coming. Yeah, please. Do you mind? There? Yeah, fantastic. So look at that. Oh, thank you. Yes, and here as well. So, thanks a lot. So this was just a simple example. It's not as easy as this. So we are working with cities actually to integrate our research into an interactive tool that they can use it. And we are helping them to maximize the transformative impact on policy, economy, and society. So thank you so much for today.
Breaking the wall of life's origin. How toxic molecules enabled the formation of RNA and early life. John Sutherland, Medical Research Council Laboratory of Molecular Biology.
On November 9th, 1989, I was getting ready to apply for my first independent academic position.
Thank you, everybody. That is a difficult act to follow. I've been upstaged many a time in my life, but never by a tree like that. So what I'd like to do is to talk to you a little bit about two big questions. These questions relate to life. The first is, how did we get here? You know, are we alone? How did we get here? How did life start on Earth, and does life exist elsewhere in the universe? And of course, when I talk about how did we get here, I'm not talking about getting a taxi from the beautiful new airport up the road there. I'm talking about how chemistry on the early Earth might have given rise to life. It's an intriguing question, one we've been working on for a while. And I'm going to call this talk basically the origin of life on Earth out of the blue, and you'll see why as I get to the end of the talk. So, of course, people have been thinking about this sort of thing for a long, long time. Humans have always wondered where they came from. And the way we think is naturally reductive. And so people in the past, in the 15th and 16th century, for example, started taking humans, normally dead humans, and dissecting them. And by dissecting them, they could work out how the various subsystems within the body worked. And so you have here some beautiful illustrations of various subsystems, the reproductive subsystem, the alimentary canal, the musculature and skeletal subsystem, and the skin. And you can imagine people at the time wondering, basically, when they did these dissections, you know, this is an inordinately complex system, the body. You know, how did it arise? Look at all these different bits, each of which is important for life. Which of them came first? You can't imagine the whole thing coming first. Something must have come first. And so you had people, basically, you can imagine the conversations. Well, the reproductive system must have come first. We all know that's supremely important. And other people saying, without food, forget it. You know, or without muscles to get you to where you want to be to eat or to reproduce, you can also forget it. And without the skin to protect you and to keep everything sort of as part of the body, again, what do you do? So people were confronted with an inordinate problem, this complex situation. How do you actually sort of imagine how it could arise? And you had to wait a while before people actually realized that the solution lay in the fact that these are organisms which have evolved from simpler precursors. And so by phylogenetic analysis, you can reduce these things in complexity, and you can see how historically they arose from simpler organisms. And ultimately, you get back to the point where you can see that all life on Earth is rooted in single cells. We think of this as the last universal common ancestor, either a single cell or communities of cells. And all of that has given rise by historical evolution to the things we see around us, the biological systems we see around us. But then molecular scientists later on start looking at this and they say, well, hang on just a minute. You know, when I start looking at this from a molecular perspective, this actually looks really complicated again, because you can't imagine a cell without informational molecules. You have to have some sort of information you can have to program what's going on in biology and also to pass the information from one generation to the next. But that's not enough. You also need to have something to catalyze, something to accelerate the processes that are going on in the cell to make them happen faster and more efficiently. There's not much point doing any of that unless you're compartmentalized. For example, if you innovate, you know, biologically speaking, if you innovate, of course, you don't want to share that innovation with your competitors. You want to keep it to yourself. And so compartmentalization enables you to do that. And finally, you need building blocks and energy if you want to do anything. You need to provide energy to drive the system, and you need to get the sort of material wherewithal to make things. And so what people are doing in the molecular sense is doing exactly what the 15th and 16th century people were doing when they were dissecting bodies. They're looking at the cell and they're saying, well, in fact, the cell itself is just too complex. It's too difficult to imagine all of that coming into being in one go. How did it start? Where did it come from? In other words, the problem reduces in a chemical sense to the fact that trying to imagine how chemistry could have produced all the individual components required to start life is just putting too big a challenge in front of the chemists who are trying to recreate this, trying to imagine how it could have taken place on Earth. Surely you need different chemistries to make the various bits and pieces. Amino acids, nucleotides, lipids, all the different components of the cell. It was imagined that you would need different chemistry to make all of those different bits and pieces. And surely if you tried to operate all those chemistries at the same time, you would get what anybody who has worked in a chemistry lab has made before, which is just an intractable brown mess. Okay? There are quite a few people who have obviously done some chemistry here. Okay? My own sort of efforts in chemistry were probably even less successful. Okay? Practical chemistry. But these are assumptions, and these have always been assumptions until the last few decades, when people have started to investigate this by comprehensive experimental investigation. And so there are people who would say, look, too complex, and so we've got to start somewhere. Why don't we start with one or other of these subsystems? So there are some people who say, look, information is more important than everything else, so let's posit that informational molecules came first. There are others who say, no, information is too complex. You can't do anything without catalysis. Let's have that. And of course, you can guess the rest. Other people who said without compartments first. And so the field became polarized, quadrupolarized, different people recognizing different attributes, all recognizing that it was too complex to imagine everything coming together at one point, but then all agreeing that something must have come first, but then differing in terms of what they thought might have come first based upon their predilections, their education, and so on, their bias. And that's kind of the point at which we got into this, because we were thinking about informational molecules. But we did have it in mind that a more holistic approach would be required than this sort of simple reductionist approach, because the reductionist approach was suffering from the fact that all of those four groups of people were all making good points. Yes, you do need informational molecules. Yes, you do need building blocks. You do need energy. You do need compartmentalization. You do need catalysis. So how do we reconcile these issues? So we started thinking about informational molecules, and we were attracted to this beautiful informational molecule, RNA, which I've shown with Jim Darnell's book cover illustration as a transition from an inorganic material, a stone staircase, into an organic structure, with the indication that if you can achieve that, the sky is the limit. Okay? So can we get from very, very simple precursor chemicals, the stone staircase, to complex organic structures? If we can, the sky is the limit. So I know it's Sunday morning, and I apologize for putting chemical structures on the slide. I will not leave them on for too long, but they will come back. Let me replace that chemical structure, which is a chain of repeating units. Okay, it's a string of beads for those of you who are not inclined to think about molecules on a Sunday morning. Let me represent it with a cartoon. And one of the things that held the field back was the fact that this cartoon reveals that the constitution of the building block, the repeating unit of the informational nucleic acids, is actually a red bit, a blue bit, and a purple bit. Okay? The red bits are sugar, the blue bits are nucleobase, the purple bit is phosphate. You learn about those in different lecture courses. Ribose you learn about in carbohydrate lecture courses, nucleobases in heterocyclic chemistry courses, and phosphate in inorganic chemistry courses. And if I had been a diligent student, I would have attended inorganic chemistry courses. As an organic chemist in Oxford back in the day, I didn't, which was, in retrospect, a mistake. But c'est la vie. So the cartoon, when you look at it that way, it basically cues in your mind a way of trying to make it. Chemists are always trying to imagine how things could have been made or how they could be made. If you've got a blue bit, a red bit, and a purple bit, how do you make that? Well, obviously, you make it by making a red bit, a blue bit, and a purple bit, and you try and join them together. And people had tried to do that for some 50 years. And it just didn't work. And so that gave people the idea that, oh, you can't join the preformed bits, doesn't work. Can't have had informational molecules, something else must have started the origin of life. That, I think, is a premature conclusion. And we thought that was a premature conclusion, so we started investigating it. And the idea was, let's not start with preformed bits. Let's start with half a nucleobase, half a sugar. The purple bit helps those join together. We can then add the second half of the sugar. We can then add the second half of the nucleobase, and we can finally incorporate the purple bit. So it's basically saying you can make things not by preforming the components, but you can assemble the components together. Now, I have a plan here to stop Klaus interrupting me with his throat clearing. So I'm going to invite him on the stage and give him a hug. Klaus, I would like to hug you. Let us hug. This is a preformed hug. And it doesn't work. What do we do? Thank you, Klaus. That was a good hug. He is, I believe, what you call the strong silent type. So there are other ways of solving problems in chemistry. So how do we go beyond that? Well, we've got to make those building blocks from something smaller. Hydrogen cyanide is where we want to start. It's a very fundamental molecule in origin of life chemistry. Turns out we can turn it into this smaller red bit. We can join red bits and blue bits. We can then assemble half sugars. We can assemble other halves of sugars. So we can do chemistry by taking hydrogen cyanide, reducing it, and turning it into the sugar building blocks. You don't need to know the details. What you need to know is that if you do that and iterate it, you can make a huge collection of molecules, which happens to correspond almost precisely to about half of the molecules you need to kickstart biology. Okay? So, sorry, the molecules you need to kickstart biology, it's about half the molecules you find in basic rudimentary primary metabolism. Half the amino acids, nucleotides, and lipids. We can obtain the feedstocks on an early planet by atmospheric chemistry after impacts, which reduces the atmosphere. And we envisage this sort of chemistry advancing in terms of complexity and indeed in the degree to which the system is alive. We don't believe in a progressive increase in complexity followed by a Frankenstein's monster moment where things suddenly become alive. We think that the various attributes which collectively constitute the living state can be acquired progressively and sequentially, and thereby you can proceed in a series of stages towards becoming alive. So there are intermediate stages of aliveness. So somewhere below that dashed line that you think of as being alive is that Berlin taxi driver who didn't get me to my hotel until almost midnight last night. Okay? There are clues that come out of this as well. So, for example, we find that the amino acids we can make by reductive hydrogen cyanide chemistry happen to correspond to half the positions in the genetic code, and they're those positions in the genetic code which are characterized as family boxes. They're boxes in the genetic code where the nature of the third base of the codon doesn't differentiate the amino acids that's coded. That, it turns out, is a huge clue. So fundamental chemistry can clue you about the structure of biology, the central dogma of biology. Chemistry doesn't just get to a certain point and then hand over to biology. Chemistry is pervasive. It reaches all the way up into the central dogma of biology. Okay? Chemistry is a powerful subject. We're in Berlin, and I've talked about the origin of life out of the blue. Why out of the blue? Well, it's because hydrogen cyanide, Blausauer in German, it dates back to the historical times in Berlin when people were trying to make pigments, red pigments. But they found that if there were contaminations in the materials they used, they got ferric ferrocyanide, a blue cyanide-containing metal complex, which they discovered could be used, and here's a picture from an art gallery up the road there, that can be used to produce a beautiful blue color in pictures. And people from that blue color, by heating it, they isolated this blue acid because it derives from a blue pigment. Blausauer is hydrogen cyanide. And hydrogen cyanide was then purified by French researchers, Monsieur Gay-Lussac in Paris. And I just leave you at this stage with a comment about what did Gay-Lussac say. He said, prussic acid, which of course was the French way of describing Blausauer, blue acid. Prussic acid, prepared as I've just described, has the following properties. It's a colorless liquid, it's very smelly, it's got a fresh taste. My God! A fresh taste. It paralyzes you, and it's a veritable poison. He survived. Anyway, so the life on Earth is the origin of life out of the blue. What about life elsewhere? Doesn't have to be blue. It could derive from being red, maybe, for example. So I'd like to ask you if you could pick from underneath your chairs just one of the 3 cubes you find there. And with Klaus's help, we're going to do an experiment which relates to the probability of life starting on an exoplanet. There are 10 to the 22 stars out there. On average, each one has a planet. My astronomer friends tell me it's a huge number. I say, that's not even a mole of planets. Chemistry, remember, is a powerful subject. But anyway, the experiment we're going to do is to try and connect these red cubes you've got, which is chemistry, with an exoplanet. And actually, secretly, I'm recruiting for the English cricket team. So if anybody can get their red cube, I'm going to count to 3. And on the count of 3, if you could throw your red cubes and try and get them in this basket, I'm going to do it too. That would be great. So, after 3, 1, 2, 3. So, is there life elsewhere? There is life elsewhere. Even without Klaus's sleight of the hand, there was life out there. Thank you very much.
No, 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 no. No, 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 no. Don't. Jürgen, too fast. Jürgen is almost all the time right, but as a physicist, time is not his issue. It's time for the family picture. And for that purpose, I'll go over there with the photographer, and please wait for the instructions. You are right in your assumptions, but I'll clarify in a minute. So everyone over here, why don't you join me? Please come up. Bitte aufstehen, and maybe you just look at me. If you see the photographer sees you. So if you want to, you may stand up. Don't forget to have one cube. One cube. I beg your pardon? If there is a state secretary around, he is allowed to come up over here, but over there he is looking nice as well. Good. Now we may need to bring this a bit more to the front. Yes. So stand there where you are. Here. Sorry? No plant, but. And if you can go to the other side. You will not be on the picture. In the photo, you have to please. With the crowd. Melania. Or join us on this side. Just next to me.
If you on the stage, could you come a little bit more forward? Yeah, like this. Okay.
Now, ready for the experiment?
We are ready, and now the instructions what to do with the
Okay. Take the cube into your right hand or into the left hand. And then we count to 3, and when it's 3, please throw as high as you can. The roof will not take off, so it's all tested.
Okay. And while you are throwing it, look to me.
Okay.
Okay.
That's the photographer.
Okay.
So.
You count or I count?
Well, that would be an interesting experiment. Who is faster? I have the microphone, so I'll do it. So we start at 1, and then it goes on to the other numbers. And when we are at 3, we throw. 3 like throw. 1, 2, throw! Now, you wonder why you have a second cube. And you are right. You may have heard about the reduction problem in science. Now we fix it here. We will do another picture.
Yeah.
But
should we try if they throw it to me?
Yeah.
I've got two.
Now the photographer has commissioned me to say you should try to throw it as high as possible on him. So again, I'm counting up. First I say 1, then I say the next number, then I say the number thereafter. And so please, 1, 2, throw! Okay. Enjoy your lunch, please.
Oh ja, danke dir, das ist
Reisschal. Ach hier. Und zwar müssen wir einmal. Das kann ich gerade machen. Okay, sorry. Aber
Es darf wirklich, also passt mal bitte auf, ja?
The thing that makes the Science Summit so unique and special for me is just the huge range of amazing people and just the really inspiring ideas that are going to change the world.
This is once in a lifetime moment for me. Yeah.
What makes the Science Summit very special and unique for me is the diversity. It's very educative, it's very engaging, and it's extremely exciting.
So for me, it's really special to meet people from all around the world, because usually it's hard to get in touch with 60 different nations.
In the pitches, we saw a lot of great ideas to shape the future on long and short term. And they address basically all the challenges we face.
The amount of innovation and the depth of the science and the entrepreneurial spirit, they're just infectious.
All aspects of science under one room in the course of a few days. You're going to discover so much. You're going to meet so many incredible people. You're going to leave feeling so energized and so hopeful.
Come here to network, come here to talk, and come here to maybe get new ideas.
We looked at 24 different ideas today. If only half of them work out, the world will be a much better place.
The Falling Walls Science Breakthrough of the Year in Life Sciences. Please welcome Jury Chair Michael Schütz, Royal Brisbane and Women's Hospital.
Ladies and gentlemen, imagine unlocking a secret which is hidden in every woman's body. So fast, so elusive, and it was so far truly never been seen. Professor Melina Schuh, director at the Max Planck, has made it possible with a revolutionary live imaging platform that captures ovulation in real time at a cellular detail. Her discovery flips decades of textbook science on its head. Ovulation isn't just controlled by the ovary. It's a self-driven, multi-step cellular masterpiece. This breakthrough doesn't just deepen our understanding, it launches new possibility for fertility treatment, non-hormonal birth control, and tackling disorders of women's health. Let's welcome Elana Shu on stage, presenting 2025 Breakthrough of Life Science, and a visionary leader breaking the wall of invisible ovulation. Congratulations, Melanie.
Breaking the wall of invisible ovulation. Our new discoveries illuminate the secrets of reproductive health. Melina Schuh, Max Planck Institute for Multidisciplinary Sciences.
On November the 9th, 1989, I sat with my siblings in front of the television, watching people cross the Berlin Wall, celebrating freedom and unity.
Yeah, thank you very much for the introduction. Ovulation is one of the most fundamental events in biology. It marks the moment when the possibility of creating a new life first begins. And yet, for most of scientific history, it has remained invisible. It happened deep inside the body, too fast to capture, too delicate for traditional methods. And because of that, ovulation was often treated like a biological black box. We knew the before and the after, but the moment itself, the critical transition, was hidden. Today I want to take you on a journey of how we made this invisible moment visible, how we uncovered the choreography behind it, and what this means for fertility and women's health. Now let me take you back in time to the summer of 2008, and in particular to the day of my PhD defense. After my defense, an examiner came up to me and said, "Have you seen this amazing publication that has just come out?" And indeed, this publication was amazing because it contained something that nobody had seen before. The first ever photograph of human ovulation. And it had been captured completely by chance. During a surgical procedure on a woman's ovary, the surgeon looked at exactly the right time. Someone in the room grabbed a camera, and with this, finally, this hidden event had been revealed. And I still remember that I found this mesmerizing. You could suddenly see here the human follicle, this balloon-like structure, and emerging from it, a golden liquid, and this contains the egg. One of nature's most private moments had been exposed, almost as if inviting us to look closer. And this sparked a question in me: what if a single photograph can already reveal so much? What might happen if we could follow the entire process of ovulation from the very beginning to the end? At the time, I was a PhD student at EMBL, a research institute in Heidelberg, and I had just established methods to study isolated oocytes, or eggs, how they are also called, on the microscope. And I could follow inside them how the chromosomes are moving, how they are separated as the oocyte gets ready for fertilization. And that was extremely exciting at the time. And I was then wondering if we can look at this with so much clarity. Maybe one day we can study ovulation as well, see how the egg prepares, how it moves, and how the follicle then transforms to release the egg. At the time, this seemed impossible. But sometimes impossible ideas stay with you quietly until the right moment arrives. Now, years later, in my own lab at the Max Planck Institute, this idea came back to me with new urgency. And we decided to attempt something very bold, to isolate the follicle and to then study ovulation ex vivo outside the body under lifelike conditions. And this was really difficult. These follicles are tiny structures. In mice, they only measure 0.5 millimeter. They are translucent and they are fragile. So if you touch them the wrong way, they will just burst. So my team members had to work really hard, and eventually they found a way to isolate the follicles with fine microneedles. And it sometimes felt like doing microsurgery on soap bubbles, and indeed sometimes the follicles behaved just like that. But after a lot of optimization, they could do it. But then the next challenge came. We had to somehow find a way to keep these follicles alive on the microscope and make sure they develop well. And this took a lot of attempts. There were days when we thought it's working, but then again, it didn't work, and we had to keep on trying new conditions. And sometimes it just felt like the biology was too difficult to tackle. But eventually, we came up with a system that worked. And you see this here. You see the lens, the objective that we use to look deep into the follicle. And then on top of the objective, there's a tissue dish that mimics the natural environment of the follicle. And inside a filter, these follicles are developing in a healthy way. And this is what we then saw. Let me guide you through this. The circular structure, that is the follicle. And the dark sphere inside it, that's the oocyte or egg. And inside the follicle, there's a lot of small cells, and they help to nourish the oocyte and to protect it. Now you can follow how ovulation proceeds. The follicle changes its shape, and then tension increases, and suddenly the egg becomes expelled. And you see this round structure on the right here, that's the egg. Now, when we could see this, the people were cheering in the microscope room. It really felt like suddenly we were able to access this hidden world, this world that had always existed, but that we couldn't access yet. And this is not only beautiful, it is also meaningful. Ovulation is surprisingly prone to failure. For instance, there is a disorder that is called polycystic ovary syndrome. And with this, many women cannot conceive, and it's actually millions of women that are affected by this. So with this system, we could now look at the motion of ovulation, and this allows us to understand better why it fails and how we might help. But we could also see more. We could look at the cells inside the follicle at great detail. And here you see the oocyte on the right-hand side, the circle, and you can really follow how it moves out of the follicle together with the other smaller cells that surround it. And if you look really closely into the circle itself, then you see pink structures. These are the chromosomes that are inside the oocyte, and they're getting ready for fertilization. And what we also discovered is that there's actually multiple different steps involved in ovulation. The follicle first gets larger, it expands, it then contracts, and ultimately it ruptures. So we wanted to find out, how is this working? How are these different steps achieved? And are they important? And we started to first look at the expansion, at the stage when the follicle gets larger, and you see this in the video, and also when you look at the volume curve, the volume increases. And you may be surprised that you may know what is already behind this expansion. It's a compound that you can find in moisturizers. It's called hyaluronic acid. So this acts like a sponge. It can absorb water and then it grows. And just like this, it is formed inside the follicle. You can see it in the center in red. And it brings the water in, and this causes the follicle to swell gently and to expand. Now, the next step is the contraction. The follicle gets smaller. So what is driving this sudden transition from expansion to contraction? And what we found is that on the surface of the follicle, there is these green structures, and they are actually muscle cells. Just like the muscles in our body, these muscle cells can contract, and they sit on the surface and compress the follicle from the outside. And this is suddenly introduced by additional hormones. They come in at just the right moment, and they help the follicle to compact. But how's the oocyte then actually moving out? And also here we were able to reveal something interesting. We marked the oocyte in green here in this video. And interestingly, while the follicle expands and contracts, it's fairly static. But then in the last moment, it gets propelled fairly quickly. And this is guided by cells inside the follicle. They change their conformation and guide the oocyte in the right direction so that it can eventually leave the ovary and be accessible for fertilization. We also looked at exactly how the follicle opens up. And what we notice when you look at this site that is about to rupture is that in the very first step, a liquid comes out, and this liquid has been marked here in red. And then after this, the first cells start to come out. And these cells are actually very important. They protect the oocyte and later the embryo as it travels through the fallopian tube. And finally, also, the oocyte emerges. So with this, we could establish a three-step model for mammalian ovulation. First, the follicle expands, and this is driven by hyaluronic acid and an influx of water into the follicle. And then it contracts from the surface through the mother cells, and this increases the pressure inside the follicle. But the follicle is not just bursting on the surface, but there is a very specific site that gets weakened. And in this way, the pressure is then released at the specific site, which is going to become the rupture site where the egg is going to emerge. So ovulation is no longer just a drawing by an artist, but we know how it works at the cellular level, and we can follow the full ovulation process live. We notice that it's a three-step process, and every single step of this process is essential. If anyone fails, ovulation does not occur, and that provides a framework for understanding ovulation disorders, such as polycystic ovary syndrome. And indeed, by imaging and manipulating the follicle outside on the microscope, we can now study the mechanism of ovulation and also why sometimes it fails. And what is also remarkable is that we can not only culture the follicle, you can also culture other tissues, even entire organs, in the same way as we did it for the follicles. For instance, we can put complete ovaries onto the filters and follow them for 5 days. And this allows us to look at steps of reproduction that were previously inaccessible. And it opens a new window to understanding fertility and women's health. Finally, I would like to thank Chris Thomas and Tabea Marks. They were instrumental in moving this project forward. I would also like to thank all the other members from my lab whose work has formed the foundation for this breakthrough. I would like to thank my institute, the Max Planck Institute for Multidisciplinary Sciences, and Göttingen University that I'm affiliated with, and also all our collaboration partners who've made this work possible over the years. Thank you for your attention. Okay. That's very kind. I hope you're up for more. This is not the end. We have something special prepared for you. You won't only see ovulation on screen in 2D, but you get to experience it in 3D on stage in an abstract interpretation. So please enjoy the choreography.
Please welcome Detlef Mennerich, Global Head of Boehringer Ingelheim Venture Fund and jury member of Falling Walls Venture.
Good morning. I can't do this. Impressive. So it's my great privilege to be here today and honored to reflect on a day on Thursday where we saw 25 amazing concept companies addressing sustainability, technology of the future, computing, and life science. And there were 25 winners, but we as a jury, and I'm on behalf of the jury of Stefan Holzbrink, we had to collect one winner. And the winner is Lumamai. And I would like to invite James to come on stage. He will address the future computing we all need, energy efficient, to address all our AI ambitions. James, the stage is yours.
The number one limitation on AI capability today is not data and it's not models or their algorithms, it's energy. We've got to the point where hyperscalers are reopening nuclear power plants just to provide the energy needed to power AI data centers. That's ridiculous, and something needs to change. So why have we hit this energy wall? Put simply, the better computing resources you have access to, the better AI capability you can build. And so historically, we've scaled our energy capability, our computing capability in two ways. One is just making our processors faster. And that means squeezing more transistors onto a piece of silicon, onto a chip. We've got to the point where there are billions of transistors on each chip, and each transistor is only a few atoms across. Again, that's remarkable, but we can't keep doing that forever. So we've gone the other way. We've scaled out instead. So we now have these data centers that have tens of thousands of processors hooked up, interconnected, to perform our AI capability instead. That's essentially brute forcing the problem. And we can't keep doing that for much longer. So we need to be smarter about it. We need to rethink the underlying technology. And that's what we're doing at Lumai by using light to perform processing rather than electronics. So that means building an analog physical system of components to perform the computation, using photons rather than electrons to encode the information. Now, there are some fantastic advantages to doing this, and it all comes down to the fact that it's an analog process, and we can fundamentally encode information as a physical process rather than turning it all into zeros and ones and shuffling it around with transistors. Now, I say the operations for AI. What do I mean by that? Well, the genius of modern AI and deep learning is it's really simple arithmetic. It's just multiplying and adding numbers, but in immense scale. So the operation that we actually need to perform for AI is matrix vector multiplication. And it's a structured form of adding and multiplying numbers together. So how do we do that using light? Well, we first need to encode the matrix, which is just a whole bunch of numbers, into a physical display, something like the screen of your phone, where each pixel represents one number. We encode the vector with an array of lasers, where the brightness of the laser encodes the number, and we pass those laser beams through the display, and that performs the multiplication. We then combine the lasers with lenses and with optics, and by combining the lasers together, we perform addition, and we then detect the output of that result and turn it back into a digital signal. That's pretty cool, and the fantastic part is that it's passive. As the light propagates through your system, you're not using any energy. So it sounds pretty amazing. It sounds like it's free computing. It's not quite that simple. We still use energy generating the light. We still use energy decoding the signal, turning it back into digital. But the 3D geometry of the system gives us a really unique property. So we want to get faster, right? Faster computing means better AI. And we do that by scaling up the system, adding more light beams, adding more pixels to our display. And as we get bigger, the number of operations we perform, the number of multiplies and adds, scales with the area of the matrix. So as we get bigger, we do more operations, and we get better AI. But the amount of energy we use scales with the input vector, which is linear. This is really unique. It means as we get bigger, we get faster, but we also get more energy efficient. That's completely unique. You don't get that with silicon, with digital, or anything else. So that's the science. And what are we actually building at Lumai? We are building AI acceleration systems based on this optical multiplier. They're completely standard form factor systems, so they look like normal compute servers, normal compute racks, but they have this optical multiplier at its core. And based around that, we can achieve massive speed up compared to digital systems, whilst only using a fraction of the power. And that really is what we need to overcome this energy wall. These are clearly artist impressions. The technology is real. So in the coming months, we'll be releasing our first demonstration system, which consists of two key parts. So one is this actual optical multiplier itself. So it's a system of optics, lenses, displays that performs this matrix vector multiplication operation. And the second is custom electronics, digital and analog electronics, which is responsible for pushing data through the optical multiplier and interfacing with the rest of the system. Add some specific software layers on top of this, and we have a complete AI acceleration system that can perform some of the biggest language models to date. Now, why do we need this? Well, we've been talking with potential partners and customers across the entire AI value chain, and the message is remarkably consistent. The gap between what we need in AI capability to perform all the amazing operations and applications we can, and the performance that we can actually get from modern hardware, that gap is huge and it's growing, and there's a desperate, desperate demand for something radical to change that. I obviously need to give a huge thank you to our team. We're based in Oxford in the UK. We have a fantastic team of all sorts of different engineers that have been and built technology for new markets across precision optical instruments, AI chips, all the different layers of the software stack, and they're just a fantastic team of people to work with. I also need to give a massive thank you to our partners and our backers. We have a global consortium of investors trying to tackle a global problem, and definitely wouldn't be possible without them. Final few comments. We are growing. We still have open positions available. We're a startup, so obviously we're fundraising. If you're a VC, please let me know. If you want to find out more about whether, why energy is a limitation to AI capability, about the demonstration system that we have coming up in the next few months, or you just want to know how we do maths with lasers, please reach out to find out more. Thank you very much. Thank you. Good. If you're waiting for an interpretive dance of laser computing, it's not going to happen. Maybe next year. Thank you.
The Falling Walls Science Breakthrough of the Year in Engineering and Technology. Dear guests, please welcome Jury Chair Rokia Raslan, University College London.
Hello everyone. As we've just heard, the rapid growth of AI demands unprecedented computing power, but it also demands unprecedented energy efficiency. Professor Fang's Tai Chi photonic chiplet and full forward mode training architecture allows computing to go beyond the current boundaries of its physical limits. This extraordinary innovation brings photonic computing into a new era and was therefore selected as our Science Breakthrough of the Year. Please join me in congratulating Professor Fang on truly breaking the wall of high-power AI needs.
Breaking the wall of high-power AI needs. How an innovative chiplet can reduce AI's environmental impact. Lu Fang, Tsinghua University.
On November the 9th, 1989, I was a child in China.
Good afternoon, everyone. I'm Lu Feng from Tsinghua University in Beijing. We are living in a time when artificial intelligence is rewriting every aspect of our world, from how we create to how we think. But behind this new intelligence, there is a hidden wall, a wall of power. AI's intelligence comes at a cost, the enormous energy. The smarter the models become, the hungrier they get. That's the wall I want to break, the wall of high-power AI needs. Let me start with something we all know. How many of you have used ChatGPT in the last hour? Well, every single day, over 100 million people use it. And together, they generate 50 trillion tokens. That's 50 trillion words and ideas every day. It's a global phenomenon, a new layer of intelligence connecting us all. But very few realize the invisible energy running behind it. Just a single day, for every single day of ChatGPT operations, it consumes enough electricity to drive an electric car around the equator for 800 times, or power an average household for 275 years. It's about 2.9 million kilowatt hours. Well, if AI is our new brain, it's the one is too much. So the question is, can we make intelligence itself more sustainable? So our answer starts with something ancient and fundamental, light. We know light travels faster than anything else in the universe. It carries information naturally. It is ultra-fast, ultra-efficient, and unlike electrons, it doesn't get tired. And here we can see some photons the tiny packets of light streaming through the space. They travel, they move effortlessly with no resistance, no heat. So we can see, what if we, instead of letting electrons compute, what if we let photons light compute? The answer is absolutely yes. But how can we actually compute with light? The approach is modulation plus propagation. Light can be modulated, encoded, guided, shaped. And once modulated, it travels a certain distance. And when received, it forms a specific pattern. The pattern is the result of the computation. And as shown in the video, we can see the light actually modulates by this kind of the, the orange and white face mask. It modulates the incoming light. And we can see, and then finally, eventually, the light in the output pattern will be focused in one single point. So through all this process, there's no resistance, no heat. Just the photons flowing, computing as they go. And we name this concept light speed computing. So we can see it's actually not science fiction. It's physics in action. So up to this point, we have used the modulation and the propagation to realize a single layer of optical computing. But one layer is far from enough. How can we do? We can build architectures connecting different layers to form the networks of light. As shown in the video, those units can be stacked layer by layer and connected in parallel, and even looped end to end. And with these architectures, we can start to try to solve some relatively simple intelligent tasks, such as the character or number classification. And that is where the boundary between the optics and the intelligence starts blur. Unfortunately, these light-based networks still face challenge. That is scalability. The physical imperfections make it difficult to scale up to a very large scale and very capable architecture. So the question becomes, what kind of organization that can really make the networks to truly scale and to think? We can, let's think about the best computer we know, the human brain. It performs 5 quadrillion operations per second, but consuming only 20 watts of the power. And it's roughly about 10 AI processors with the performance, but consuming only the energy with 3 meals, just 3 bowls of the rice we can see, right? It's very tiny, right? It's very small, yeah? Now, how about today's AI? To match that performance, it probably will need 500 times more power. That's about 250 kilograms of the rice every day. Wow. Thank you very much. Thank you. Thank you. That's huge, right? So here's a question for you. How much rice do you think light needs to compute? May I? Well, just 50 grams. So, thank you so much. Thank you. Yeah. So the brain reveals something, profound. The intelligence does not have to be always, energy or power hungry. It just has to be beautifully and efficiently designed, just like our human brain. So what we can learn from that, the existing, neural networks, they have a potential to scale up by stacking layers by layers. Does such kind of the electronic-based neural networks always work for the photonics? Unfortunately, the answer is negative. In the optical systems, the physical error accumulates and propagates when we go deeper and deeper. And it will, make the, it will actually try to make the position and the robustness to be a big issue. So, such kind of the, electronic-based deep architectures can hardly, be scaled. Interestingly, the brain research reveals something really useful and meaningful, the shallow brain hypothesis. Our human brain does not rely on many deep layers. Instead, we will try to distribute the massive computing in a parallel and shallow pathways. So inspired by this, we designed the Tai Chi, a tiny lighter brain. In Tai Chi, we will not try to stacking layers in the deep way, but we will try to distribute, the very complex tasks in a parallel and a broad way. So it's a kind of a physical embodiment of intelligence. There is an old proverb in China, and 3 ignorant cobblers add up to a genius. And it perfectly describes the distribution concept behind the Tai Chi triplets and architecture. In Tai Chi, we will try to distribute, a very complex and large-scale computing task into many simple ones. And we design such kind of top-down architecture. And we can see with this kind of the architecture, for example, for the multi-category classification, we can try to decompose it into a series of binary decisions, and they vote together to get final results. And for the generative task, we can divide a big image into several patches, and each decorated with its own stylized texture. And finally, they come together to get the original image as if painted by Van Gogh or Monet. So, this is elegant and efficient. So the benchmarks are astonishing. Compared with the conventional processors, Tai Chi can run 100 times faster. It's like taking a train from Berlin to London, a journey that usually takes 10 hours, right? And now it could just take 6 minutes. And besides that, Tai Chi consumes 1,000 times less energy. Imagine charging your mobile phone once, and instead of lasting for just 1 day, and now it keeps running for 3 years. So it's the AI that is high speed, high fidelity, and high efficient all at once. And imagine what happens if this computing power moves to the forefront. The drones can think, the autonomous robots can work seamlessly with the humans, and the autonomous vehicles, they can perceive and decide in the light speed. And the ChatGPT-like intelligence can run directly in your mobile phone without relying on the cloud computing. And that's intelligence, not just the things that we connect to, but it's something that surrounds us, sensing, adapting, learning at the speed of light. And that's the future we are stepping into, a world that light itself empowers intelligence. Beyond the performance, there is something deeper. Our goal is not just to make AI more efficient, but something more meaningful, from efficiency to the effectiveness. It's the idea we name as regenerative green AI. So the intelligence not just sustains the world, but actively restores the world. AI that gives back to the society, to the planet, to our shared human story. And that's the journey actually we are on. if we can make, the machines to think in light, perhaps it can think lightly on the energy, lightly on the resources, and gently on our Earth. Thank you very much. I would like to express my deepest gratitude to the Falling Walls Foundation for the great honor they have given me, and to Tsinghua University for providing a home where science and imagination can meet. And to my lovely and wonderful students, they are the true geniuses behind this work. And it is their passion and perseverance that can make light, not just a computer, but inspire. So let's make intelligence truly intelligent. Thank you so much. Thank you.
Please welcome Ilan Shem, representing our partners for the Women's Impact Award category.
Women and girls make up half the world, and yet face systemic changes every day across every field. And so we felt, the Elsevier Foundation, the Volkswagen Foundation, Falling Walls, we felt it was high time to celebrate the research that was uncovering blind spots and really tackling long neglected areas. And so the Women's Impact Award, we're delighted that this year's winner, Dr. Angela Gonçalves, has done groundbreaking work in women's health. So she is an AI scientist who's become a molecular biologist, and she has integrated AI, molecular biology, and clinical insight to understand how reproductive cycling affects aging and disease. Now, you have to think about areas like endometriosis, ovarian cancer, menopause, long neglected and not well understood areas of women's health. So her pioneering work tackles gender bias in medicine and redefines the science of healthy aging. We bring you Dr. Angela Gonçalves.
Breaking the wall of healthy aging in women. Our menstrual blood is revolutionizing medical diagnostics. Angela Gonçalves, German Cancer Research Center.
On November the 9th, 1989, I had just started school in Portugal.
Thank you very much and good morning. Today I want to talk to you about menstruation. But don't worry, I've replaced all the blood in my slides with blue liquid. It's funny, isn't it? We can have all the blood we want in movies, but mention period blood in an advert and suddenly women are secretly bleeding in shades of blue. This is silly, so I'm switching to red. So we often treat the menstrual cycle as an inconvenience to be scheduled around. But with this talk, I want to convince you that it should be a front and center issue. Picture the uterus on an ordinary day. It's a thin, neat neighborhood of cells. 2 weeks later, this neighborhood is 10 times bigger. Blood vessels have carved roads through it, immune cells have moved in, and all the cells are now doing something else differently. They have all changed jobs. 2 days later, this neighborhood collapses. Half the cells are dead, and an inflammatory cleanup crew has moved in. So you build, remodel, demolish, and repeat every cycle for decades. But the menstrual cycle doesn't just impact your uterus. It affects your ovaries, cervix, vagina, and breasts. It impacts your skin, your immune system, and your body temperature. It influences how fast your heart beats. Mine is beating very fast right now. Your mood and your metabolism. When was the last time that your physician asked you what day of the cycle were you in when you had that cholesterol test? Never, and yet it matters. Despite being essential to half of humanity, we still don't have clear answers to many basic questions about the menstrual cycle. For example, what is the cellular program that triggers menstrual bleeding? Why do cycle lengths vary so much from person to person? Why do some women experience a lot of pain and others none? Why do some women bleed a lot and others very little? What is the causal link between obesity and disturbed menstrual cycles? What causes what? What is endometriosis, really? And my personal favorite, how does a lifetime of cycling affect your aging and your long-term health risks? 1 in 3 women suffer from menstrual abnormalities, and yet the investment into these issues remains strikingly low. How low? I would like to try something out with you. So you're probably sitting on these big pieces of paper that are on your seats. So I'd like to ask you to hold them up for everybody to see. Okay, thank you. So each one of you represents an interventional clinical trial in Europe at the moment. Now, can I ask the ones with the white papers to put their hands down? The ones with colored papers are the only ones that are working on women's specific issues. So these 20 have a very heavy burden on their shoulders because they alone are working in all of these things. All of the gynecological cancers, so ovarian, cervical, vulval, breast cancer, and they're also working on infertility, pregnancy complications, contraception, incontinence, menopause symptoms, gynecological infections, pain, female alopecia, endometriosis, fibrosis, adenomyosis, the list goes on. So that's 20 out of 400. That's the level of attention that's being given to these issues. This is an important health gap and something that an opportunity to help a lot of people. So for the last 10 years, I've been working using cutting-edge molecular and computational techniques to study women's health issues. And today I want to tell you 2 stories, one about cancer risk and one about menstrual blood itself. So I'm a cancer researcher, and naturally I'm particularly concerned with ovarian and endometrial cancers. These cancers tend to be diagnosed very late in life, 15 to 30 years after menopause. And yet, one of their most important risk factors happens decades earlier, and it's the number of menstrual cycles you've had before you hit menopause. Epidemiological studies give us this statistic. Each additional year of cycling adds roughly 9% of uterine cancer risk. How much is that? Is that a lot? Let me put this into perspective for you. So an average Western European woman will start her periods at around age 12, will have 1.8 children, will spend 4 months without periods because of breastfeeding, and will reach menopause at the age of 50. So that adds up to 450 cycles in her lifetime, which is equivalent to 34 years of cycling. Now picture a hypothetical hunter-gatherer woman in the Paleolithic. She will start her periods later, she will have more children, and she will breastfeed them for longer, so that she accumulates only about 160 cycles in her lifetime. That's the equivalent to 12 years of cycling. That's 22 fewer years of cycling than an average woman today. So average women today have 2 times the risk of this Paleolithic woman, and the women that are at a higher risk these days have 5 times the risk. Now here's a mystery. Why is it that having more cycles leads or is linked to having more cancer? Is it because of repeated tissue injury and repair? Is it ovulation itself? Is it hormone exposure? We don't know the molecular details of this. And finding out, that's not just a curiosity. That's an opportunity for prevention. One reason we don't know many of these, we don't have answers to many of these questions, is because we scientists tend to spend most of our time looking at disease and very little time establishing healthy baselines. It's like we're trying to design a marathon training plan by only interviewing the runners that collapsed at kilometer 10. So we've been using a different approach. So instead of chasing disease, we've been modeling healthy cycling and aging in humans and in animal models. So we've looked across tens of individuals. We looked at hundreds of organs, thousands of images of histological structures, and millions of single cells using machine learning and AI to sift through billions of gene expression data points. And here's what we found. So we found that cycling generates an acute burst of inflammation, which is what we expected to see. But then, contrary to the tidy textbook story where endometrium is supposed to heal scar-free, we found that inflammation in the endometrium and fallopian tubes does not reset to zero after each cycle, but it accumulates, accumulates fibrosis, which you can think of as microscopic scarring. We then stop cycling in young animals, and we let time pass. And the result was striking. By old age, we found that 80% of the scarring we could see was due to the cycling, and only 20% was due to aging. So our hypothesis is that repeated cycles create this chronic inflammatory and fibrotic environment that accelerates the tissues towards carcinogenesis. And if this is true, then anti-inflammatory strategies, some of which are already being used for prevention in other diseases, could be tailored for chemoprevention of gynecological cancers. So studying healthy aging can be tremendously valuable for prevention. But how do we move on to do bigger trials more broadly in humans? After all, we simply cannot go around collecting invasive biopsies from healthy young women every month, right? So unless we use something that is already leaving the body. And I'm talking here, of course, about menstrual blood. So menstrual blood isn't just blood. It's a mixture of blood cells and blood plasma, but also cells from the endometrium and their secretions. Now, 10 years ago, we asked ourselves, can we use this blood for easily, at scale, in real life, for research and diagnostics? So we started running studies with healthy volunteers. So in the very first one, healthy volunteers self-collected menstrual blood samples using menstrual cups, and then they just deposited them in our institute's special boxes we had installed at our institute's toilets. That's it, so no clinic visit, no invasive procedures. They simply dropped off their samples, and the research team never even saw them. So back in the lab, we then measured how much blood can we get, how many live cells are in it, what cell types are in it. And we measured how much do these vary from person to person, and how much does it vary for a person from cycle to cycle. But most importantly, we wanted to know how do the menstrual blood samples compare to endometrial biopsies at the molecular level. And so we've measured both, we integrated them computationally, and we found that they line up very well. So in other words, menstrual blood is a realistic stand-in for an endometrial biopsy, which of course opens up very exciting possibilities, for example, in diagnostics. So moving on to applications in diagnostics, I'd like to say a few words about endometriosis. So endometriosis is a chronic inflammatory disease that is characterized by the presence of lesions that look like the endometrium, but are in the wrong place. So instead of being inside of the uterus, they show up on the surface of the ovary, for example, or of the bladder, or of the intestines. It's typically very painful. Women with endometriosis often tend to bleed a lot. They often struggle with infertility, and it's a risk factor for ovarian cancer. The WHO estimates that 1 in 10 women suffer from endometriosis, which makes it 3 times more common than dementia. And yet it currently takes 7 to 9 years for a woman with symptoms to get a diagnosis. So we asked ourselves, can we use menstrual blood to help with this? Okay. So to find out, we enrolled a group of volunteers with endometriosis, and we found gene expression patterns that separate them from healthy controls, which now paves the way for us to validate these findings in larger scale studies. But the potential of endometriosis doesn't end here. Of menstrual blood doesn't end here. Menstrual blood is already being used for monitoring diabetes, and there is proof of principle that it can be used for HPV screening. And I'm also very excited because menstrual blood has the potential to also extend access to healthcare for women that live in areas of the world where invasive pelvic exams are still a barrier. So as you leave this venue today, I want you to think differently about menstruation. We need to stop treating menstrual biology as an afterthought. This is not a niche. This is essential for the well-being of half of humanity. The return on investment of solving issues like endometriosis and other menstrual abnormalities is enormous, with the potential to save billions of years that are currently being lost to pain and disability throughout the world. I also want you to think about these healthy references. If cycles leave fingerprints, then... Oh, thank you. This is going to make me waste even more time. Then those healthy references are essential for prevention. So if you are a funder or policymaker, please support and fund healthy references for women. These are essential for prevention, but are missing. And if you are a scientist or clinician, include menstrual cycle phase into your experimental design. I know that we're still struggling for I know we're still struggling for having trials report gender disaggregated data, but I'm telling you what needs to happen next. We need to start including cycle phase into our designs. Can I take it?
And please welcome Urmila Chadayamuri from Nature.
Good afternoon, everyone. So, put your hand up if you have ever wanted to be an astronaut. Yes, I thought so. In fact, in the last ESA call for astronauts, there were more than 22,500 candidates, right, of whom just over a dozen made the cut. And we are very fortunate to have one of those people here with us today. Matthias Maurer will be joining us on stage after a couple moments. And we want to talk today about, well, what is it that fascinates us so much about going all the way up there, but also what it takes for us as an international community of individuals, very committed individuals, scientists, researchers, HR personnel, international collaborators, educators, to get this to happen. How do we work on these ambitious projects together? And we're going to have a really enlightening perspective from an astronaut right here today. With that, let's get started with a little movie of Matthias's experience in the Cosmic Kiss mission recently. welcome on Earth.
Thank you very much.
So let's get started. I find it remarkable the number of different scientific experiments that you carry out in space. You are a scientist by training. Can you tell us a little bit about your personal research background?
So I'm a material scientist by education, and when the wall came down, I was actually doing a civil service also as an ambulance driver, and this means like I have specialities in material science, but also later on I worked in the development of medical disposables, and this covers a lot of the research areas that we are doing currently on the International Space Station. We do a lot of material science development. We are having, as humans, being research subjects as well, but we're doing also like fluid physics, chemistry, and biomedical experiments in space.
It's really remarkable. How do you go through this process of experiments being proposed? How are you trained to perform such a variety of things up there?
Yeah, so here we come to a point where a lot of people believe astronauts are superheroes and that we are actually the scientists who develop this stuff. No, we are just like the technicians. The smart people are you guys on the ground. You develop that stuff, hand it over to the space agency, in our case to the European Space Agency, send it up to space, and ideally it's you with the space experts have developed a procedure that is fail-proof so that we don't mess up your experiment.
Have you ever messed up an experiment?
No, but this is actually the biggest fear of an astronaut. It's like there's so much research and so much time spent in the preparation of an experiment that, like for some people, it's almost a decade that I have worked for an experiment. And so once it up is in space, we as an astronaut think like, okay, I absolutely need to make sure that I don't mess up a very important experiment.
So a lot of mental power, a lot of focus, don't mess up.
Absolutely.
Yeah. Another thing so you're cooperating with the scientists on Earth who have, on the ground, so to speak, who have developed these experiments, designed them meticulously to make them as fail-proof as possible. You're also collaborating with all the other astronauts in your crew. To what extent, in fact, do you think the selection process has to do with your technical skills versus your interpersonal skills and ability to live with such a diverse crew of people in a small environment for an extended period of time?
Yeah, you touch on a very important point. It's the social skills of an astronaut is actually the one that is most decisive during a selection process. Especially, I mean, we as Europeans, we don't have our own access to space, so we're always like either flying with the Russians or flying with the Americans. I also trained in China because at a certain point in time we were looking forward to flying to the Chinese space station. And so you need to be open-minded. You need to be prepared to learn different languages when you want to become an astronaut. And you also need to be aware of the cultural differences. And so different people from different cultural areas tackle the same problem on a different way. Mm-hmm. And this can be so enriching, but if you're not aware of this, it can also be kind of a block to a useful or meaningful cooperation.
Mm-hmm. Just a moment to brag about Matthias's skills. You speak English, obviously, and Deutsch, and Französisch, and Espanol, yes, and Ruski, yes, and Ruski. Mandarin.
And Mandarin I learned, yeah. It's like saying speaking, it's like I learned all these languages and I don't even speak perfect German, I have to admit.
But do these come in handy on the ISS?
Yes, it is. It is. Like speaking languages is really important, and it's an enabling factor.
Mm-hmm. So, and you've also studied, trained, and worked in a lot of different countries. Can you tell us some of the places that you've been and the sort of cooperation that has enabled this?
Yeah, so I was very lucky that I could participate in different studies in Europe. So first was like as an engineer, I wanted to learn English, so I studied in the UK. And then a new combination studies was offered to my university, material science, a European degree. So allowing me to study also in Spain and in France, having also to do international training in Latin America. After my studies, I was also in Korea. And then later on, during the astronaut training, I was in Canada, in Japan, also in China, and in the US and Russia.
That is so many places. It makes International Space Station, it gives a whole new layer of meaning to that. I'm just curious, during your education in France and Spain, was that pre-EU or post-EU?
That was already in the EU.
That was in the EU. Yeah, so that definitely facilitated your ability.
It was when the UK was still in the EU.
Right.
good old times.
Yeah. Great. Now, scaling back a little bit, back when you were still in Germany, you are from Saarland, is that correct? And I actually found it really fascinating that you come from a family of mining workers, isn't that right? So how did you get it into your head that you could go all the way up there, 400 kilometers above the biosphere, really, where humans have barely been? What was that? What is this path to your envisioning this dream and the steps that you took to get there?
Yeah, so I come from a rural area, and there is it's very far away from NASA, from astronauts, and so what happened there is at that time was during the Cold War. We had lots of jet flying, practicing like dogfights at very low level, and so I thought like, wow, being a pilot. Having a look at the Earth from the third dimension, this must be fabulous. So my dream was to fly and to become a pilot. I never thought that I would become an astronaut. Actually, it's like I limited myself in dreaming big. I thought like astronauts are superhumans that live in the US. And so I saw them on TV and I thought like, this is so far away, don't even start this dream. And I think
Space or the US?
It's like in the US. It's like yeah. Don't even start this dream to become an astronaut because that's only possible for Americans. Yes. And so it's the biggest burden or the biggest obstacle of like getting closer to this dream was actually my mindset. And this is my key message to the kids. It's like Europe happens everywhere, and the future happens everywhere, even here in Europe. And you need to dream big. Only if you dream big, you can make your dream come true. So my dream to become an astronaut only happened when I was an adult. I came back from work in the evening, I switched on the TV and I saw in the news the European Space Agency is looking for astronauts. And I was already like 37 at that age. And so
I thought the age limit for applying?
Exactly, yes. Exactly. It's like in Europe you can only once in a decade you have the chance to participate in an astronaut selection. So either you're selected or the dream is over. So it's a once in a lifetime chance.
And you went for it. Yes. Oh, wow. We have so much time. I'm so excited. Okay. So now we can dig into the juicier details. So the Cosmic Kiss mission. How long were you in preparation for this? What is the story behind it? And also, why is it called Cosmic Kiss?
Yeah, so a spaceflight mission, a long-duration spaceflight mission, usually lasts for 2.5 years of preparation, 3 years. Depends a little bit on like all the elements that you need to train for. I had some additional elements because I was preparing for a spacewalk with the Russians on the Russian side and also with the Americans on the American side. So you need to learn 2 different systems. That makes the training a little bit longer. The Cosmic Kiss name of my mission is a name that I chose, and it expresses like the declaration of love to space. When people ask me like, why should we fly to space? ESA gives you a justification, which is like, if you invest €1 into space, you will get back €2 as an economic benefit. And people look at me and think like, really? I never got back €2. So it's like, this is an explanation. It might be economically correct, but it's not appealing. It's not. It doesn't like throw people off the chairs and say like, yes, let's do more space. So it's like I want to motivate people by the dream. Everybody of us has this dream to explore areas that we haven't seen yet, and flying to space, flying to the moon as the next destination, and then going even further to Mars. And then this huge universe, there's so much to discover. Actually, 95% of all that out there, we don't even know how it works. And I mean, imagine all the dreams that can come true, all the science, all the knowledge. It's just fascinating. And so that's why we need to fly to space.
Okay. Admission, I did not want to be an astronaut. I'm an astrophysicist.
I have a deception for you. I believe you are an astronaut, because everybody of us is an astronaut. We all live in space, and we kind of tend to exclude. We tend to think of like Earth is not part of the universe. Because when people tell me like he's been to space, I'm like, you have been to space. We are in space right now.
That's amazing perspective. However, something that you said in another interview that I found really beautiful was that your first moment of looking at the Earth from space, you described the atmosphere of the Earth as like a delicate soap bubble that was wrapping the Earth, and that everything that had ever happened in the history of humanity had happened within that little bubble, right? We did not evolve to withstand cosmic radiation, right? We did not, you talked about, maybe you can tell us a little bit about all the things that are peculiar about living in space that the body is not designed to do, and how your body adjusts while you're there and when you come back. And also another thing that I think we forget, you are sort of a master of perspective, right? Astronauts are masters of perspective in this sense. You see things from a different scale. The ISS is about 400 kilometers above the surface of the Earth. The moon is about 400,000 kilometers away from the Earth. Mars, right? I mean, like, so can you just talk a little bit about this? What are the things that you mentally, physically have to get used to when you're in space, and how would these things change as we get more ambitious and think about missions to Moon or Mars?
Yes, so when you prepare for a space flight, you train a lot, you try to understand everything that might happen, can happen, and how everything works. And so in your brain, you are fully prepared, you have everything understood. But then you fly out to space and you open the window shutters and you see for the very first time planet Earth from space. And that's a moment that gives you goosebumps. It's just like within the blink of an eye, you understand everything that you've understood in your brain, also in your heart. It's so emotional. It is, you see planet Earth, a blue marble, and that's the place where humans can live and thrive without any additional protection like a spacesuit or a spacecraft. It's just a perfect place. And then around planet Earth, there's just black, cold, black emptiness. And you think like, wow, there's nothing, there's no plan B. Not that I can see it from there. It's like everything is down there. Everything belongs to this one planet. You fly around in 90 minutes. So like every day I see 16 times the sunrise and sunset. And then you see a volcano eruption, and the huge smoke that comes out of this eruption goes half around the planet, and you understand, okay, this might, if things like that happen even stronger, this might affect the climate. And it's not only on that place where the eruption takes place, but also on the other side of planet Earth. So our planet is an entity. It's a spaceship. It's the best spaceship that we have with 8 billion astronauts, if you want to be an astronaut or not. And we as astronauts in space, we know our spaceship only works if we work as one crew, as one entity. Mm-hmm. And then you look down at planet Earth and you think like, what are you guys doing to your spaceship? It's just like, we need to tell them like, work together. It's like, stop killing yourself, stop destroying your spaceship, work together. There are enough challenges that we need to tackle.
Yeah. We are coming to the close of our conversation, so couple last questions. What are the walls that you would like to see falling down when it comes to space exploration and when it comes to transferring knowledge that we accumulate on space missions on Earth?
So I want us to look not only horizontally, but also up to space. Space is the next domain that comes really, really of importance. In the past, only professional astronauts could fly to space, and so it was limited to space agencies. But now we have a democratization of access to space. More people can fly to space, and it's not only for research. We will also start production in space. So medical components, medication tests can be run in space. People are also dreaming of creating organs in space, so that could help people on the ground. But then also high-quality optical fibers that are 1,000 times purer than optical fibers that we can produce here on the ground. And so this might help for computing, optical computing, but also for medical components for analysis. But then Also, we heard about the energy needs for AI. Some big companies are actually investing in space to bring data centers up to space, because there you can produce the energy, solar energy. You have more intensity. You have it 24 hours a day. And also the vacuum of space, it's cold, so you also can have already the cooling included. I'm not sure if that is the best way to go forward. I like saving energy much better. But it just, like, these are some small examples to illustrate that space has so much to offer, so many chances, not only on science, but also on economy. And we need to be prepared and need to like take up the challenge to conquer space, yeah.
Yeah. Matthias Maurer, everybody, thank you so much.
Thank you very much. Thank you very much.
Thank you.
Thank you.
Urmila, you left one question open.
Ah, yes.
What did you have for lunch in space?
Well, it's like you get space food prepared, but sometimes you have a special day with a special dish. And so I wanted to make this an endeavor, my mission, where a lot of people can participate. And so I had my common people from my fellows from Saarland region to select my space food, so that I had a little bit of like my home area back in space. So yeah, that was a very delicious homegrown space food.
Now, probably the people would like to know how looks food from Saarland when it comes to space.
It looks like Berlin food in space. I believe it all looks similar.
Klaus will show us wonderful Saarland food in space.
It's a can. It's a can. Or it might also be a pouch. So this is space food, and you just need to warm it up because we don't have a kitchen in space. We don't cook there. We just eat the stuff that is good for 3 years. It's been prepared on the ground, and it tastes tasty. If you have no alternative.
Some of the food in space was left over. And we asked, why can't we have it? And so if you wish to, you can have Saarland food from space now for lunch. We had an argument, should there be a free choice for you or not? And the minority prevailed, saying you should have a choice. So when you come down, you see a picture, and we may see it now up here as well. Can we have the picture, please? Yes. So you can opt for the line of the, as you know, the line is the real place to be. The line of the Thuli are exciting, cool people. They line up for this. And if you are in the other line, you are very welcome as well. So enjoy your lunch and see you back in a couple of minutes. Thank you. Thank you. Dankeschön.
Good morning, Berlin.
To a celebration of contemplation, of innovation, and of collaboration.
Falling Walls is so special because I get the chance to meet people that I've known for years online, and it's such a unique and unbelievable opportunity in such a lovely city as Berlin.
I never thought I would make a lot of friends that are, like so passionate about science in different fields.
You have people with a more entrepreneurial background, you have VCs here, you have scientists. There's a big combination of different backgrounds, and I think that's what really makes it interesting here.
Is this unique multicultural experience that we have here.
And it's the passion the scientists show in the various projects they are in charge of.
I feel the vibe, it's the community that is really making the effort on science and science education.
It gives me a sense of inspiration.
To give voice to young people with groundbreaking ideas.
So seeing the diversity in the number of the engaged pitches, seeing the audience how loving and embracing they were.
Because they continue sharing their stories, taking pictures, sharing contacts.
It's impressive to know that there are so many scientists, so many researchers involved in making the future better.
Some strong statements were made, some optimism about the future.
The walls have to fall not just physically, but also mentally, to basically think out of the box.
Thank you also for your support.
Please welcome Jürgen Mlunek, Chairman of the Board of Trustees of the Falling Walls Foundation.
So, hello everybody to this afternoon session. How are you? Are you energized? Very good. How was the food? Let me see. Who took the astronaut food? How come that this is only this half of the room? Okay, some over there too. Did you like it? I must say, okay, but this was less than before. Anyhow, so I think this must have been an interesting experience. Wonderful. We start the final round this afternoon. It's the fourth day. Why 4 days? Because normally it's 3 days, but the Stifterverband and some other organizations asked us to have an additional day back-to-back for the Summit for Research and Innovation. And we will hear a little bit more about that in just a moment. So the first day of Falling Walls is the day of pitches, as you remember. This is the lab competition, 100 young people from all over the world, 3 minutes each. It's the venture competition, it's the engaged competition, and the female science talents meeting. And what we are trying to do right now in time, 2 minutes each, is to give a short review of what happened. And I start with Annie. This name is even more complicated than mine. Annie Mamushashvili. Okay, from Georgia. So, you are one of the lab participants. How did you like that day?
Thank you. I'm a participant from Georgia. I feel extremely lucky to be here and participating as a pitcher. We not only got 3-minute professional video to which reached the wide audience, but I also felt I was seen and I was heard as an inventor. And I got the huge motivation to come back with even greater achievements to share in the future here, and I can't wait that to happen. And the people that I've met, they are so inspiring, so kind, and so helpful. And I've learned in these last 3 days about me, my weaknesses, which became my strengths in communication, because it's very difficult to overcome your fear of not being enough as a scientist, as a person. And thank you so much.
Thank you, Annie. And we hope that Georgia stays with Europe. Okay. We continue with the Summit for Research and Innovation. One of the organizers, Georg Schütte, will give a brief summary. Georg, why don't you come up for a moment?
Well, we had one day of debate to talk about time to act for greater competitiveness in Germany and in Europe. The Federal Minister of Research, Technology and Space was here, and she reminded us that we in Germany and in Europe need to escape the middle technology trap. So we do not need incremental improvements of existing technologies. We have to be more daring. We have to overcome complacency, self-centeredness in Germany, be courageous and move into deep tech innovation. She announced the high-tech agenda, and we heard about it this morning again. Germany is going to devote 18 billion euro to new technologies. What do we make out of it? We looked into the structure of the German science and research system, and there were two keywords. We need focus and division of labor in order to move ahead. And number two, we need more agility, speed, and we need to create networks. Networks across disciplines, networks between sectors, public and private, and we need to network across national boundaries. And that was our second session. How do we collaborate internationally in a fragmented world? One of the keywords is research and knowledge security, new boundaries in a free exchange of knowledge. And the questions are, how do we further collaborate across the Atlantic with countries like China, with new partners in India? How do we reach out to other continents like Africa, like South America? So these were challenges which we tried to address. And we not only talked about research security, we also looked into security-related research. And that was the last session, innovation for security and innovation through security. Amongst others, the director of the Los Alamos National Lab was here, and he explained that the concept of security reaches from Earth systems research, shrinking ice shields, and new ways to move for submarines and vessels around the world when the ice shield shrinks, all the way to AI in technology that we need for defense purposes. And I learned a new term which is software-driven defense. We had the research head of the German cyber agency here who explained about new concepts of defense technology. If I sum it up, there were three keywords that stick to my mind: speed, scalability, and impact. And we have to work on all of it with your help next year and continue the debate.
Thank you very much. Georg, I think doing this together, Falling Walls and the Summit for Research and Innovation was a win-win situation. You can come again. Okay, we had another event on November 7th together with one of our partners, the Boston Consulting Group, and Johanna Pütz tells us a little bit about the outcome with respect to sustainability and climate. Johanna.
Thank you. Thank you. Indeed, it was an enormous pleasure for us to curate 3 hours of intense discussion about climate and sustainability. For BCG, I mean, we're not so close to science on a day-to-day basis, so it was definitely a first for us to be present at the stage at Falling Walls. And yeah, we curated a journey that started with science. We had Johan Rockström, who gave a very impressive speech, as always, of course, when Johan speaks. Then politics with Mr. Frank Wetzel, outlining the agenda of the federal ministry when it comes to climate and energy, as well as the economy. And then lastly, we had 3 panels with corporates and also startups. We had ENBW, Scilab, Mercedes, DHL, and many others talking about how they take all of these insights and convert them into action. And for me, there's actually 3 lessons. Number 1 is, especially from the Rockström speech, we all need to do better in educating people around us about tipping points in climate. I feel like so many people that I speak to in the educated circle in Berlin think climate change means they are going to be able to grow wine in Berlin. And that's just not the case. It's tipping points and it's existential threats, and we need to bring this message and educate people and really break through this wall in thinking. Thank you. Then second lesson, maybe a more positive one. While I think in general companies can do more and are not yet doing enough, I think we also have many positive examples of people who are spirited, who are passionate, and who are very impactful in driving change, both when it comes to growing new technologies and also transforming, which is actually much harder, transforming large corporates and the organizations that they are in. So that made me hopeful in our discussion. And then the third point, maybe on a macro level, I think in Europe we have all of the conditions to be successful. We are not fully seizing them right now to our greatest opportunity. So what we need in Europe is a coherent plan to scale and to invest in green technologies and the green transformation, and then also the capital to back that up. Both are still not here in a sufficient manner, and we need to work to getting that. Thank you. Thank you.
Coming back to this sustainability and climate issue, I must say I was really moved for a moment when I was listening to Maura, our astronaut. When he said, we are all astronauts, I mean, I never really thought about it. There is no planet B. Okay, cut. We have one more person coming on stage here, Efrosini Ciliotti. Efrosini, welcome. Efrosini is heading the Department for Strategy in the Ministry for Research, Technology and Space. What is your take, you know, as an administrator and shaper of the future in the ministry out of the conference so far, Efrosini?
Thank you, Jürgen. Hello, everyone. Well, BM-FTR, or BM Future, as our minister called it, has been proudly supporting the Falling Walls Science Summit from the onset, and it's been wonderful to see how this format has developed over the years. It has literally put Germany and Berlin, of course, on the map. I take 3 learnings from the past 2 days who have been extremely inspiring and insightful. The first is that the German government and BM-FTR more specifically are, or seem to be right on track with the high-tech agenda. We seem to have identified the right priorities for the next couple of years. The second is we have an excellent research base. We have amazing minds, not only in Germany, but in Europe and beyond. And we need to continue investing in platforms such as this one, where an exchange without mental blocks or restrictions is possible. And the third thing is something that also Matthias Maurer talked about this morning, mindset. we need to be bold. We need to look at the blind spots that we ourselves leave behind. And in order to secure our democracy, our resilience, but also our technological sovereignty, we need to be daring in getting scientific results faster into industry, into technological results and products, because otherwise we will be left behind. So thank you again for your contribution today, and thank you for creating this platform and bringing it to Berlin every year.
Thank you. Thank you, Frosini and the Ministry for the support, the long-standing support. We really appreciate this. So, I have a few minutes left. Who of you loves to dance cha-cha-cha? I talked to my wife before, and she said, "Don't make a fool out of you." So, anyhow. So, next year. Why cha-cha-cha? I don't know. Maybe some of you know the cha-cha-cha theory of discovery. Who knows that theory? Okay, it was published a couple of years ago in Science, as a matter of fact. Cha-cha-cha is charge, challenge, and chance. Charge is to solve the most obvious unsolved problem. The classical example is gravitation. You know, the apple that fell on the head of Newton, and Newton thought, well, how come? And he said, I have to find this out. So that's charge. Challenge, the classical example is Niels Bohr. A lot was known about, you know, the nucleus and the electrons, so there was a lot of information, but somebody had to put it together to come up with an explanation. So that's challenge. Now, chance is Röntgen. Probably many of you, also the non-physicists, know perhaps that when Röntgen was doing experiments with cathode rays, there was a photoplate nearby, and his wife had the hand before the photoplate. And then when he developed the plate, he saw the hand bones of his wife. And then he said, something is going on here. I have to understand this. And this has to do with serendipity and something that people call a prepared mind. Now let me ask you, coming back to charge, what do you think are problems related to charge? What are the most obvious unsolved problems, let's say in science? It's a science conference here. Me as a physicist, as one example, I would say dark energy, dark matter, as an example. What else? Come on. Okay, can you?
So I think for the whole summit, it's really been explicitly mentioned by many people over here. But really, women are really underrepresented in research and science.
One more. There must be more obvious unsolved problems. If not, this would be paradise. That can't be.
So we have internet now, right? But I'm dreaming of a quantum internet in the next decade.
Okay. Very good. So, what about challenge? Me as a physicist, okay. Good. Anyhow, so it's really important, and that's my message. You know, discoveries, you have to work on them. You have to be, you know, sensible. If an apple falls on your head by chance, think about the most obvious unsolved problem in whatever topic and whatever discipline you are in. And with respect to chance, have a prepared mind. If we have a prepared mind, I think there will be a bright future. So there is the final stage now open to this afternoon, and I think we just start. Thanks for being here. Keep a prepared mind. It's wonderful to have you with us here at the Falling World Science Summit. Thank you.
The Falling Walls Science Breakthrough of the Year in Art and Science. Dear guests, please welcome Jury Vice Chair, Christina Landbrecht, Schering Stiftung.
Very happy to be here. Marco Barotti is a media artist, and he's also the winner of the Art and Science Breakthrough Award 2025. Barotti combines biology, music, sound, and robotics. And currently, for the past 5 years, he's been experimenting with sound in coral reefs. Those reefs, as you may have heard, when a report was recently published by the Potsdam Institute for Climate Impact Research, are at risk of dying out due to global warming approaching critical tipping points. Barotti's project, Coral Sonic Resilience, takes a stand against this. The sound of a healthy reef is played to a damaged one, and as if lured by a siren song, fish and other sea animals follow the choral melodies and return to repopulate the reef. We, the jury, consider this a breakthrough. And on behalf of all the members of the jury, I congratulate Marco Barotti for winning this award.
Breaking the wall of ecological healing through sound. How underwater installations can revitalize natural environments. Marco Barotti. Marco Barotti Studio.
On November 9th, 1989, I was a small kid in Italy. I remember watching Roger Waters' concert The Wall on TV with my brother and feeling something huge was happening. Who would have thought that years later I would end up living here?
This was the sound of a healthy coral reef, alive with crackles, pops, clicks, and low-frequency hums made by fish, shrimp, and other marine organisms. These sounds are not just beautiful and cosmic, they are vital. Young corals and fish larvae, they use sound to navigate and to locate suitable habitats to settle and grow. So in other words, if a reef is alive with sound, it's most likely to stay alive. But when reefs degrade, they grow silent. Coral reefs are fundamental to life on Earth. They provide incredible benefits to human and more than human communities. They offer food, shelter, ingredients for pharmaceuticals, carbon storage, and much more. They support the life of 1/4 of marine species, even if they cover only the 1% of the vast ocean floor. They are incredible, aren't they? Unfortunately, the reefs are in crisis. Around 50% of the world coral reef, they have been lost or severely damaged. And about 90%, scientists says, it could disappear by 2050. Coral bleaching, which is happening when stressed corals expel the algae that live in symbiosis with them, is becoming more frequent and severe. So these impacts are not just single events anymore. They are part of a larger pattern that shows us how climate change and human activities are pushing ecosystems beyond their limits. So protecting corals is not just an act of conservation. It's an act of care, reciprocity, and justice. When we safeguard coral ecosystems, or any ecosystem, we also nurture the intricate web of life that binds us all together. I have a background in music, and sound has always been the core of my work. For me, it's not just a medium, it's a way of sensing, connecting, and understanding. Through sound, I try to open a space where data becomes experience, and where listening becomes a form of awareness, engagement, and repair. I create interventions in urban and natural environments. They are data-driven, kinetic sound sculptures that resemble animals and plants and deal with environmental and social issues. In every work, I try to recreate the biology and the functionality of specific species in nature through technology. I collaborate closely with scientists, and I use environmental data sonification to bring critical topics to the audience in a more tangible, sensitive, and poetical way. And today, I'm here to share with you my new artwork. Coral Sonic Resilience is an interdisciplinary art and science collaboration rooted on acoustic ecology. It explores how the soundscapes of healthy reefs can actually support the recovery of damaged ones. The work builds on the research of Professor Timothy Lamont from Lancaster University. In the Great Barrier Reef, Professor Lamont applied a technique called acoustic enrichment. He recorded the soundscapes of healthy reefs in multiple locations, and then he played those sounds back into degraded colonies using underwater loudspeakers, like that one. Over 40 days, his research proved that fish and other marine organisms came back to the degraded colonies because of sound. I got fascinated with the concept of acoustic enrichment and by the idea that sound can actually heal an ecosystem. Well, more than fascinated, I would say I got obsessed with it. And I tell you why. In my previous work about corals, I developed what I call techno-shamans. Techno-shamans are algorithms trained on shamanistic music from South Korea, South America, and Japan. The aim of the artwork was to create speculative healing rituals for corals using bleaching and ocean data sets. It was an artistic exploration to heal the reefs through sound. So when I found out the scientists were doing in real life what my speculative artwork was trying to do metaphorically, a new path opened up. I started diving into acoustic ecology, asking myself, how can sound and art become an act of repair, contributing directly to ecological recovery? So for the first time, I made a work not for humans, but for fish and corals. Okay, here we go. Thank you. Thank you. Okay, the applause in the middle never happened, so thank you very much. I was very convincing until now. Let's see what I do now. So the sculptures are 3D printed using ceramic and calcium carbonate. They are materials which reflect the natural composition of corals. The design and textures is based on 3D scans of bleached corals and is thought to invite marine life to shelter, to settle, and grow. This photo is taken in Feridu Island in the Maldives, where together with the University of Padua, I went to record the soundscapes of healthy reefs and to install the sculptures in damaged areas and coral nurseries. Over several weeks, we collected different type of data, and we ran many tests to optimize both the sculptures and the solar power station. The floating solar station is actually providing clean and autonomous energy. It captures sunlight through photovoltaic panels and converts it into electricity to power the sound system and so the underwater loudspeakers. The buoy is designed in Berlin and is thought to fit inside a small suitcase to be easily transported and installed anywhere and to enable sound installation in remote marine environments. The soundscapes were recorded in 4 different healthy reefs around Feridu Island. And in order to portray, let's say, a healthy marine environment. After the installation and after the recording, we put together sort of, how do you say, a picture of the soundscapes of the healthy reefs of Feridu. Now that we are ready for the new setup, we are going back to the Maldives in February 2026 to install new sculptures, and a marine biologist on site will then collect coral and fish growth over several months. And a scientific paper will be published by the end of 2026. Beside the Maldives, the project will go to Jamaica, to Bahrain, and hopefully in Singapore. So now, coral sonic resilience is evolving into a body of work, right? Parallel to the underwater artwork, I'm developing new iterations of the project for art exhibitions above water for human audiences. The journey starts right here at Falling Walls and Science Week, and it will continue to national and international venues at ZKM in Karlsruhe, Art and Science Museum in Singapore, Science Gallery in Melbourne, and Ocean Space for the Venice Biennale of Architecture in 2027. And whether the artwork is installed underwater or in a gallery, it is driven by the same purpose: to change our perspective, to invite new ways of listening, and to engage with and to protect the fragile ecologies we are part of. In this way, coral sonic resilience is not only exposing a crisis, it takes part in it, intervenes, and proposes new models of coexistence and regeneration. Thank you.
Breaking the wall of mapping the body. How global efforts are unlocking the blueprint of human cells. Stephen Quake, Stanford University.
On November 9th, 1989, I was in my 3rd year of undergraduate studies in California and read about the wall falling in the newspapers the next day.
So we're going to talk today about atlases, which are collections of maps, and journeys of discovery in which such maps are made, both literal and metaphorical. In 1831, Charles Darwin left England as a naturalist on the HMS Beagle on a journey which he voyaged around the world. He, as a naturalist, was in charge of collecting specimens everywhere they stopped. The journey they thought would take 2 years, it took 5 years. 18 months of that was at sea. He was seasick the entire time. But everywhere they stopped, he collected specimens. And scientific discovery is such a wonderful process because it's such a chance-related process. We heard about that from Jürgen a moment ago. You often drill dry wells. Occasionally you find something. In the central section here, if you all look under your seats, a handful of you will make a discovery. Here we go. There's one. There. An illustration of the diversity one finds in nature when you go looking. When Darwin stopped, when the Beagle stopped in the Galapagos Islands off the coast of South America, Darwin noticed on these islands there were finches that had beaks of a variety of shapes, which had, sort of seemed to be tuned for different purposes on different islands. And it wasn't until he got back to England, he collected them, didn't realize until he got back to England that they were different species, and through that observation, others discovered the principle of evolution by natural selection, which changed our understanding of the world and our place in it. Amazing as it was, Darwin was missing an important piece. He didn't understand the mechanism by which information was transmitted through generations and altered, and we'll come to that in a moment. Last year, I had a chance to pay my own personal homage to Darwin by joining a boat, the Oosterschelde, that was sailing to recapitulate his journey. It took more than a year and a half, and I joined as a volunteer crew member for 2 weeks on a journey in the middle of the Pacific there. We sailed through the Tuamotu archipelago, the entire breadth of Polynesia. It was 1,200 miles. We had to crew the boat. We had to take the helm, keep it on the proper compass bearing, which is much more difficult than it sounds. We had to raise and lower the sails, which we had no winches for. It was all done with ropes and pulleys. I got to be very good at pulling rope. That was my skill on the trip. And, I wanted to sail through the Tuamotus and see all the coral atolls there. But the captain insisted we sail around and only enter at the very end. And the reason for that is the Tuamotus were not charted to sufficient precision to take the boat there. And it was amazing to me to learn that there are parts of this earth that are still uncharted and still amenable to human exploration and discovery. And I feel like science is the same way. For young scientists, it's not all been discovered. There's a bunch left to do. So let's return to Darwin's missing piece. We now know that it was DNA. That is the molecule of inheritance. It's the sort of mechanism for information storage and transmission. You may have noticed, Jim Watson died last week. He was very famous because along with him, Francis Crick, Rosalind Franklin, they discovered the chemical structure of the DNA molecule. They didn't discover there was a molecule of inheritance, though. Okay? Anyone know who that was? More than a decade earlier, Oswald Avery discovered DNA was the molecule of inheritance, and it took a long time to figure out its structure. The structure led to many other important discoveries. So DNA is often talked about as the blueprint of the organism, but it's really not. I think that's an incorrect metaphor, because every cell in your body has the same DNA molecule, the same DNA sequence, yet the cells that derive from that are very different. A heart cell, lung cell, neuron, all very different from each other. Sidney Brenner pointed this out 20 years ago. I think a better metaphor is that the genome is the parts list. And the discovery I want to talk about today is how we went about trying to work out how those parts were used to make all the different cells of the human body. It's, even 20 years into the genome revolution, it's amazing to me that we cannot predict the cell types of an organism from the genome alone. It just hasn't been possible. And so we had to go and set about and do that experimentally. And the experimental method we used was called single-cell transcriptomics. I'll talk about that in a moment. We've made these maps. I can say we're comfortably now in the state of having very good rough drafts. It was a huge team science project. Each of these papers had more than 150 authors on it. I had to found and lead a new research institute called the Chan Zuckerberg BioHub to do it. We did this not just for human, but for mouse, fly, lemur. These were the first whole organism cell atlases. Let me tell you a bit about how we did it and a little bit about what we learned. So it was a decade-long journey to make these atlases. It was a further 2 decades before that to build the technologies to do that, and I contributed in part to that by the development of microfluidic tools that allowed one to automate and do large-scale experiments in biology. We did sort of the earliest version of this in my lab. They went on to become very important commercial tools that enabled the field. Like many other discoveries in biology, the creation of these atlases rested on the development of new technology. So Watson was a famous showman. He said he discovered the secret of life. The second secret of life was discovered by Jacob and Monod. They discovered that some genes control other genes. And the way that works is they will encode for proteins that actually bind to the genome, they bind to the DNA, and recruit the machinery that copies the information into RNA. Those are called transcription factors. There are 1,622 predicted transcription factors in the human genome. Some have been very well studied. Others are only predictions based on their sequence characteristics. We decided to take that whole set of sequences and look for whether they were expressed in our human cell atlas, in our Tabula sapiens. And indeed, we found all of them. And it was amazing. This was a new map. A map of which transcription factors determine which cell types. You can see it here. Each column is a different transcription factor. Each row is a different cell type. And for many of these transcription factors, they're expressed in only one or a handful of cell types and really define their state. On the right of the figure, you see transcription factors that are universal. They're expressed in every cell type of the body. They're ancestral, inherited through evolution, used not just in humans, but in many other creatures. Some of them are very well studied. They control cell division, stress response, metabolism. We discovered new universal transcription factors that, despite more than a century of cell biology, still had not been found. And this is now a map to understanding some very important biology that they regulate. Another discovery we made with the Tabula sapiens was that about senescence. So a man named Hayflick discovered something called the Hayflick limit, which says that cells cannot divide indefinitely. They can only divide about 40 or 50 times, then they stop. When they stop, they enter a new state. He called that senescence. It's something we now associate with aging. We discovered, and there's been wonderful work on that over many decades. We discovered that within the Tabula sapiens, we had the largest collection of human senescent cells ever found. And so we decided to study their gene expression programs across different tissues and discovered that there's not just one type of senescence. There's several different types, a set of different transcriptional regulatory programs that are used in different patterns depending on which cell type and tissue. And that's going to send us off in, I think, new understanding of how aging works. And finally, the connection to human health and therapeutics. Using the techniques that we devised to make these atlases, we've used to also discover new drugs which are now being tested in humans. In the one case, biologics being used to cure food allergies by a company called IGeneX. In another case, small molecule drugs against a protein called KDM4, which is a very important cancer target. And we'll see how all that turns out. Let's return to Darwin and evolution. How do we interpret the information in the atlases? It's fairly challenging, and at a point I decided the way to do this would be to build large language models on the atlas data and to use that to try to find insight. The same kind of models that are used to make ChatGPT and are driving the AI revolution, but instead of training them on text, we would train them on the molecular counts that were in the cell types that were in the atlas. And we decided to do this not just for the human data, but across evolutionary time, to try to have the models learn intrinsic aspects of evolution. So the current model is called Transcriptformer. It's developed in collaboration with Theo Karletsos and his group at CZI. It's trained on 12 different species, so using not just our data, but other data generated by the community, both in human and in other organisms. And we did it in such a way that it's trained only on sort of a matrix, There's no biology put in the model, only the molecule counts for each cell type, and done in a way that we can do it independent of genome for any creature. This has been very powerful. We've seen some incredible emergent behavior come out from that. For example, when we use the model to analyze data that it was not trained on, for example, data that was used to sort of simulate infection by stimulating cells with something called lipopolysaccharide, and we use the data from cells from one species to try to predict what happens to another species, the model can do that, and it does it very well. This is part of the value of having a model trained across different organisms, that you can see what happens in, for example, animal data and try to predict what might happen in humans. Or you can predict what might happen in disease. So the model was trained on healthy cell data. We used it to test it on data that was taken from COVID experiments, where some cells were infected with a COVID virus, others were not. And could the model distinguish which were sick, which were healthy? It could do that surprisingly well. Finally, these models are a long way from perfect. And the last two slides here, I just want to illustrate that. The first with some data. So we began to test the models on data that was not part of the training from completely alternate species: lemur, frog, lamprey, coral. The model is able to classify cell types from those organisms, and therefore, it's got great value. But you can see the performance is decreasing the further away you get from human evolutionary distance. We don't know why. That's something to solve yet as we continue to work to refine them, but illustrates kind of the frontier challenges of the model. And last, I will leave you with a story. This is Jorge Luis Borges. It's a short story. This is the entire story. It's a one-paragraph short story, and it's about map making. And I often get asked, you built this model only on RNA data, and you treat this cell as a bag of RNA. What about the structures in the cells? What about the proteins? What about the metabolites? And I would say, yes, all those things matter. Depends on what question you want to ask. And models someday will incorporate some of that information. But the point of a model is not to make an exact replica. And that's the point of the story here. It's the story of an empire that tried to build perfect maps. When it was so perfect, it was exactly the same as the city. And it was useless at that point. Thank you.
The Falling Walls Science Breakthrough of the Year in Physical Sciences. Please welcome Jury Chair Ian Wormsley, Imperial College, London.
So it's good to have Darwin precede you on these things. Well, it's great to be here on the anniversary of the falling of the Berlin Wall, an event of really major significance globally, of course. And I want to now turn to the Physical Sciences Breakthrough Prize. And I want you to imagine what it would be like to be able to talk to someone halfway across the world, knowing that you can't be eavesdropped upon without knowing that that has happened. Quantum makes that possible, breaking the wall to communications whose security is guaranteed by nature. Now, I want you to imagine further that you can do this in space, so you really can have global reach in that conversation. And putting quantum communications links on satellites breaks through that wall to enable us to talk across the world in a secure way. And the prize winner for this year, Professor Janwei Pan, led a team that actually has achieved this, breaking through that wall, what, as William Shatner has told us for many years, the final frontier, to get ourselves into a position where quantum really enables new ways of thinking about how we communicate with one another. We'll hear a video from Professor Pan in the moment, who unfortunately couldn't join us today, but his colleague, Professor Dr. Cheng Peng, will be accepting the award. Thank you.
Ladies and gentlemen, Thanks to the organizer for allowing me to participate in this grand event via pre-recorded video. Quantum communication offers unconditional security in principle, yet extending its range has been a significant challenge. We have demonstrated global quantum communication through quantum satellite missions. To achieve high-efficient global quantum communication, the construction of a quantum constellation is necessary. Now we have taken the first step by realizing real-time quantum communication using a microsatellite and a mobile ground station. In the future, we will launch more microsatellites and strengthen collaboration with colleagues from all over the world to jointly build a global secure quantum network.
Breaking the wall of practical satellite quantum key distribution. How advanced light technology is revolutionizing secure communication. Cheng Jipeng, University of Science and Technology of China.
Thank you, everyone. Actually, my English is terrible. But don't worry, my smart glasses are here and will save today. So, security has always been at the heart of the human civilization. For security, people built the castle in Germany. People built the Great Wall in China. But sometimes people broke the wall to gain freedom and security in Berlin. So, long before the internet, human has already cared about keeping information safe. In ancient Greece, Spartans used a wooden stick called a scuttle to hide secret messages. In ancient China, generals used hufu to matching tiger-shaped tokens to confirm that military order were real. Today, information security protects almost everything we do: our money, our communication, our personal data. For example, the RSA encryption system is based on math mode, a math problem that it is easy to do one way, but extremely hard to do reverse. So, with this method, two communicating parties can share a computed result to create secret keys and encrypt message and verify identities. This idea is so clever and is so useful that almost every people in the world are using it. So, but there's still a problem. This kind of security is not fair to everyone. Why? Because the reverse solving is very hard, but not impossible. So we can see the RSA being corrected time and time again. So who has the ability to correct this? Of course, is the strongest government, the richest company, or the most talented hacker, not us, not ordinary people. Moreover, in 1990s, scientist Peter Shaw showed that a quantum computer could one day crack RSA. Now, only a few places, like the United States, like Europe, like China, or some big companies like Google or IBM, are able to develop such computers. This makes the digital world even more unfair. Everyone, no matter who they are, rich or poor, strong or weak, should enjoy the same right to information security. So, how can we make this possible? We need an unbreakable encryption system to achieve true digital equality. It is the final goal of the information security for many years, but it has never been achieved. Quantum mechanics gave us a new solution. About 30 more years ago, more than 30 years ago, some scientists proposed a new method called quantum key distribution. Unlike ESA, which generate key by sharing the communication result, QKD shares quantum states, such as a single photon or entangled photon pairs, to generate the unconditional security key. So, quantum mechanics tell us that matter or energy have their smallest unit for light, that is photon. It cannot be divided further. Quantum mechanics also tell us that a photon can exist in superposition, meaning it can be several states at the same time. Because of this, we cannot simply copy or clone them. So, based on this, quantum key distribution can never be broken, and can never be broken even with the powerful computer or quantum computer. But this is the first QKD experiment. Because of the quantum signal, like photon, is very weak. The first QKD experiment center key just 32 centimeters. So how to extend the QKD to long distance is the most challenging problem. About 20 years ago, Professor Jianwei Pan returned to China and focused that challenge issue. I was very lucky to become his PhD student. This is Professor Jianwei Pan, and in the middle, this is me. As you can see, my hair is all black at that time. Back then, we were thinking, when the light travels, photons are always lost along the way. So, how to make it possible, how to make it work around the whole world? The answer is outer space. Beyond the Earth's atmosphere, even photons that have traveled millions of light years away from the stars can still reach us on Earth. So, using the quantum satellite, we can greatly reduce the loss of photons and make global quantum communication possible. With this dream, we start our more than 20 years research. To reach outer space, the first step is to pass through the atmosphere. It has an effective thickness of about 10 kilometers. Sorry. In 2005, we completed the first quantum entanglement distribution over a distance equal to this thickness. It was the first time that a quantum entanglement was observed over 10 kilograms, a very large scale at that time. Later, we developed the QKD. We developed the QKD with decoy state method, which ensure the security at long distance. So, with this, this will help us to turn our dream into reality. Next, we began to consider sending or receiving photon between satellite and Earth. It is not only a scientific challenge, but also a space engineering challenge. So we wait a while. So we did a series ground tests in the Qinghai Lake to ensure the success. We carried a serious ground test at Qinghai Lake in China. We used the 100-kilometer free space link to simulate the high loss between satellite and ground. We also use a moving platform like balloons to simulate the satellite motion. Based on this test, we achieved a quantum entanglement and key distribution over 100 kilometers. Because Qinghai Lake is one of the most beautiful places in China. We set up our equipment on the island in the middle of the lake. The view was amazing. Local people and the llamas, and especially this young llama, often comes to see our experiments. By the end, we become good friends. Based on this effort, in 2016, we launched MISSUS, the world's first quantum science satellite. With MISSUS, we first implemented satellite to ground QKD, and the distance extend from the first experiment. Remember, the first experiment 32 centimeters to over 1,000 kilometers. Thank you. Moreover, using the entanglement source in the satellite, we sent entangled photon pairs to 2 ground stations. In this way, the satellite don't know nothing about the key, even if the satellite is controlled by your enemy. Once the entanglement is verified, the security of the QKD can still be ensured. So, we use the fiber-based QKD networks to connect the nearby cities, and we use the satellite to link distant ones, such as Xinjiang, Vienna, and Calgary in Canada. So far, we have realized the dream we had 20 years ago. Quantum communication can be applied everywhere and can provide unbreakable keys to everyone. Following the MISSUS satellite, many LEO quantum satellite projects have been proposed worldwide. We hope all satellites can work together to form a uniform network, more secure and not depend on one provider. So, making the satellite smaller and cheaper will become the key direction of the future for the future. In the past years, we developed the G91 microsatellite, reducing the payload weight from 100 kilograms to 10 kilograms. And the ground station, the ground was reduced from over 10 tons to about 100 kilograms. It can generate about 1 million bits of keys per orbit in real time, making QKD more practical for applications. We're also working with the international colleagues through the International Telecommunication Union to promote the standard of the quantum communication, aiming to make it more practical, to help QKD work together with today's communication system and become a key part of global communication networks. Of course, the foundation of QKD actually lies on the long-distance transmission of quantum states. So this technology can help us transfer the time and the frequency. We have achieved over 100-kilometer high-precision transfer and now expanding it to satellite. It will promote the groundwork for the next generation definition of the unit of seconds. Thank you. Beyond the human-generated quantum states, quantum signals, we are also studying the natural photons from distant satellite. Welcome. By performing a kilometer-scale optical signal we observed that star, right here. This is about 600 light years away from us. That is the result. So that's all. Thank you.
Please welcome the chair of the Emerging Talents Jury, Wolfgang Marquardt, retired chairman of the board of directors of Forschungszentrum Jülich and VP of Helmholtz Association.
It's my pleasure and honor to briefly introduce you to the Falling Walls Lab, a full day of 100 pitches, 3 minutes each. Exciting pitches, inspiring and truly impressive. The topics really covered almost all the societal challenges we face. Energy, environment, climate, circular economy, health, and what more. It was also science communication at its best, really at its best. All the presenters did a great job in presenting their work with great clarity. The presentations were well structured and extremely well presented. Creative ideas we saw, how to explain a complex topic to a broad audience in a short period of time. Simple language, animations, clever illustrations, and also analogies were used in these presentations. At the end of the day, I was really leaving with a very positive perspective of the future. I'm sure and reassured that the future will be bright in face of these brilliant talents we saw. After the pitches, the jury had to solve a difficult problem, selecting 3 winners from, I would say, 100, almost winners, which we saw during the day. And you now will enjoy the presentations of the research topics of the 3 winners in the sequence, 3rd, 2nd, 1st. Enjoy.
Breaking the wall of rare disease therapies. Harry Wilton Clark, Falling Walls Lab, Edmonton.
Today, I'd like to talk about rare disease. But first, I want to start by introducing a patient I work with, Riley. Riley is a 12-year-old boy with Duchenne muscular dystrophy, a disease causing muscular weakness and breakdown. Riley likes to watch hockey, he likes to play with Lego, and sadly, Riley is unlikely to live past his 25th birthday as his heart fails from his disease. Sadly, the story of Riley is far from unique. Duchenne muscular dystrophy is just one of about 7,000 different rare diseases affecting patients worldwide. And while these diseases are individually rare, collectively they become incredibly common. Currently, 1 in 15 children are expected to be born with at least one rare disease. Despite the debilitating or even fatal natures of these diseases, however, the development of therapies to treat them has lagged far behind for a few key reasons. Firstly, these diseases have very small patient populations. This makes it hard both to study and understand the disease, as well as to meet regulatory requirements when building therapies to treat them. Secondly, therapy development is expensive. It takes an average of $172 million to develop a single drug through FDA clinical trials right now. When we think about 7,000 different rare diseases, each with small patient populations that offer low ultimate return, there's low financial incentive for large corporate investment in developing therapies for these diseases. As a result, the development of therapies to treat rare disease has lagged far behind our clinical understanding of the same diseases. What we really need is a generic approach that can treat multiple rare diseases simultaneously. That's where our group comes in. Our group has focused on the development of antisense oligos, or what we like to call genetic bandages. These bandages bind to a mutation in a patient's genetic code and hide it from the body, helping to treat disease right at the genetic level. One of the main benefits of this approach is the flexibility. The same bandage with slight tweaks in its targeting sequence can be used to treat different mutations causing the same disease, or even different diseases entirely. And this approach is theoretically applicable to up to 80% of all rare disease right now. Another big benefit of these bandages is the safety. Many of you might be familiar with other gene therapy modalities, things like CRISPR. An issue with CRISPR is that the safety profile has been inconsistent between trials right now, limiting the speed with which we can develop these therapies. For genetic bandages, the safety profile has been very consistent, leading to less legal red tape and a streamlined FDA development platform that leads to faster, cheaper development of therapies when using this chemistry. Touching back on Riley. For my PhD project, I actually partnered directly with Riley and his family, and together we were able to develop bandages capable of treating his muscular dystrophy. Through this, we were hoping not only to help Riley and other young boys with muscular dystrophy, but to establish genetic bandages as a foundation for treating rare disease. There are over 7,000 different rare diseases, and 95% have no treatment, but we're looking to change that one bandage at a time.
Breaking the wall of solid tumor shields. Elena Barba Sarasua, Falling Walls Lab, Spain.
The clock is ticking and cancer is winning. Every year, 20 million people are diagnosed with cancer, which is almost 1/4 of the population of Germany. And every year, 9.7 million people die of cancer, which is almost 3 times the population of Berlin. But why is this happening? Because conventional treatments like chemotherapy and radiotherapy are not working. Almost 40% of the patients relapse within the first 2 years of treatment. So we turn to immunotherapies, where we have bispecific T-cell engagers that form a bridge between the T-cells and the tumor cells, enabling these T-cells to kill the tumor cells. And then we also have the very well-known CAR T-cell therapy, which are engineered T-cells that can recognize and kill the tumor cells. These immunotherapies have shown impressive results in some blood cancers, like leukemia or lymphoma. But in the case of solid tumors, they fail in 80% of the cases. So it is urgent to find an alternative for these patients. And that's where we come in. We're engineering T-cells to stab cancer. Stab T-cells act as living drug factories, as they are continuously secreting bispecific T-cell engagers. So they act as effectors, but they also recruit bystander T-cells, thus massively amplifying the immune response in the tumor site. Take multiple myeloma. Today, the standard immunotherapy treatment is CAR T-cell therapy. And yet in mice, they seem to control disease in the beginning, but in the long term, they end up relapsing. Now, can you imagine what happened when we treated these mice with a stab T-cell therapy? They are 100% cancer-free. We published this work in Science Translational Medicine, and we worked really hard to patent this strategy. And do you know the best out of it? That we're treating our first 15 patients in the upcoming months. So this is getting real, and now we're scaling it to solid tumors with a multi-target strategy. For that, we're engineering stab T-cells against surface antigens, but also against intracellular antigens that are expressed on the surface. And on top of this, we're engineering these stab T-cells to secrete NK cell engagers. So just by modifying a few number of T-cells, we're boosting a local immune cascade that stabs and destroys the tumor. And while the clock is still ticking, we are all in a mission to go from this to this, stabbing cancer. Thank you.
Breaking the Wall of Molecular Monotony. Karen de la Vega Hernandez. Falling Walls Lab, Marie Skłodowska-Curie Actions.
When I tell people that making medicines is like ordering pizza, I get that look. But I've tested this theory. Hear me out. One night, my friends and I tried to order a pizza with all our favorite ingredients. As you can imagine, the pizzeria said, no way, only one topping per pizza. That same "you can't" is the wall we hit in chemistry when we try to make medicines better. Think of paracetamol. It's great for headaches, but too much damages your liver. Now imagine a safer version, a paracetamol 2.0. How do we get there? It's like making the perfect pizza. We start with the original base and make small changes until we find the best one. The problem is developing that better medicine requires hundreds of versions, but we cannot make them. Most tools only let us create one new version. Pepperoni? Fine. But pineapple? We need a different tool or start from scratch. It's slow, expensive, unsustainable, and a bit heartbreaking, because we know those better versions could exist. So it's time for a new recipe. My job is to create tools that let us make as many versions of a medicine as we need. This new compound is our tool. It's like a multi-plug adapter. We plug it into a medicine, and it gives us 3 sockets ready to attach any topping we want. Now we can make hundreds of versions from the same base. And we've already used this tool to modify medicines for hypertension, diabetes, multiple sclerosis, and more. With a robot, we made 24 versions of one medicine in just one day, instead of weeks. And the beauty is that 80% of all existing medicines are already compatible with our adapter. So this one tool could help improve many of the medicines we all rely on. But that pizza night wasn't over. Just as I hit order, a friend shouted, "Wait! I'm gluten intolerant." Now the problem is the crust. And in medicine, sometimes the crust, the core of the molecule, isn't good enough. But changing that is much harder, like making your pizza gluten-free after it's baked. Impossible, right? Our second tool makes that possible. It edits the core, adding an extra carbon atom, creating a new base we can then decorate to make many more versions. Tiny tweaks can spark big changes. Our tools make those changes possible. Because every wall we break in chemistry isn't just molecular, it's human. It's a wall between disease and cure, between what is and what could be. This is our slice of that future, our contribution to making medicines better for everyone. Thank you.
The Falling Walls Science Breakthrough of the Year in Social Sciences and Humanities. Dear guests, please welcome jury member Yasemin Soysal, Berlin Social Science Center.
Yes, walls are broken in social sciences and humanities too. From Uganda to Sierra Leone to the Americas, Professor Miriam Dino's work breaks the wall of silence, accountability, and passivity that surround children born of war and violence. Her work reminds us that stories of trauma can also be stories of strength, and that sometimes a drawing, a photograph, or a mask speaks truth more powerfully than any word can ever could. By training war-affected youth as co-researchers, Miriam turns research into collaboration and participants into agents of change. She fuses rigorous social science with humanities, with profound humanities. She redefines what it means to do ethical, transformative, and courageous research. And for that, the jury unanimously and enthusiastically honors her with this award in recognition of her scholarship that produces excellent knowledge that heals, empowers, and transforms lives. Let's celebrate her and her work.
breaking the wall of war and its impact on children. How conflict shapes the lives of the next generation. Miriam Denoff, McGill University.
On November the 9th, 1989, I was in high school, and I remember seeing images of the wall falling on television.
It's an honor to be here. For the last 25 years, I've been conducting research on children and families affected by war. Early in my career, I've been looking at child soldiers in Sierra Leone, and more recently, looking at children born of war in northern Uganda. What got me started on this journey are images like these of children caught in conflict, and particularly images like these. And what was always striking to me is that we get very little understanding of the child behind that gun. How did that child pick up that gun? What happened to them afterwards? Where are they now? And what is their story? And we see a unidimensional portrayal of that child as dangerous and threatening, but the complexity of his life is lost. As we all know, armed conflicts around the world are having devastating impacts on children. And violations against children are increasing, not decreasing. And when we see statistics and numbers, they are vital to understanding the realities of children, but they don't tell us the whole story. And when children's stories are told, they're often told from the perspective or through the eyes of adults, through researchers, through humanitarian workers, through family members. But rarely do we get to hear from the children themselves. So the goal of my work is to gather the voices and perspectives of children affected by war and to ensure that these perspectives are heard and that their voices are heard and valued. So as a result, because ultimately their stories matter. And yet, gathering children's stories of war are incredibly difficult to do. How do we ask a child to recount what has happened to them in some of the most difficult and violent times of their life? And as researchers, we can actually do great harm. So some of the challenges involved are asking a child to recount can actually end up in re-traumatization or re-victimization. And for example, a young girl that I had met in Sierra Leone had been abducted as a 9-year-old. She was forced to participate in the war and remained captive for 5 years. During that time, she had basically came back to her community, and she had 2 children that were born of rape. How do we begin to ask that child the circumstances of her life? Another challenge is that in many cases, words alone can't adequately capture the horrors and the complexities of war. And yet another challenge is that children traditionally in social science research have often been regarded as objects of research rather than active agents in their own lives and circumstances. So I needed to find solutions to these challenges. And over the years, I began to use 2 approaches. One, using art, and the other, using participation and bringing young people onto my research team. And with these approaches, they help to address some of the challenges, and they also help to address breaking down several walls. One, the wall of silence, that through art and participation, that we can ensure and gather the voices of children so that they are heard and valued. And we can also help to break down this idea of the passive child, that involving children in research and drawing on their expertise can ensure that they're not regarded as passive victims, but active agents in their own circumstances. And finally, the wall of inaction, that by bringing forward their stories, that we can ensure that their voices are heard and that change is possible and that we go beyond complacency and move beyond outrage. So I'm going to talk to you a little bit about these two approaches and how I use them in my research. So in terms of art, art offered multiple ways for young people, particularly children, to express themselves. That children don't have to talk. They can paint, they can draw, they can make a mask, they can write and perform plays. So they're enabled to go beyond words. And this process itself can ensure that they go beyond words, that this process also can be healing, it can be empowering, and therapeutic, and also, importantly, fun. But each technique that we use must be in line with the culture and context in which we're working. In terms of participation, young people are the experts in their own lives and circumstances, and we need to be drawing on this knowledge and expertise. Young people who have lived through conflict have a tremendous amount of knowledge. And so, for example, young people, in many circumstances, when they've become a child soldier, they have been leaders to other young people in an armed group. We need to be tapping into that leadership. So what I do is that I train young people, and after extensive training, they eventually become co-researchers on my research team. And after extensive training, they collect data, they analyze data, and they disseminate information through, for example, community conferences. Now, in the spirit of gathering stories, I want to provide you with the stories of two young people affected by armed conflict. And one story is going to establish and illustrate the importance of art, and the second one is going to establish the importance of participation. So Millie is from northern Uganda, and her story is going to help us understand through her art. In northern Uganda, there was a 20-year civil war. And during that time, the Lord's Resistance Army, or what I'm going to call the LRA, was trying to overthrow the Ugandan government. And during that time, committed horrific atrocities against civilians. And as part of those atrocities, the LRA abducted an estimated 60,000 children into armed conflict over a 20-year period. Now, to understand Millie's story, we need to understand the story of her mother. Millie's mother was abducted when she was 12 years old by the LRA. She was held captive for 8 years, and during that time, she was forced to become a fighter. She was forced to marry a commander, and she gave birth to 3 children who were born of sexual violence, and Millie was one of those children. Now, Millie grew up in an armed group. Her early years were in the LRA. And eventually, she was able to escape with her mother and her 2 siblings. And when I met her when she was 15 years old, she could be considered one of the lucky ones because she was able to attend school. But Millie she faced major challenges in her life because she was born of sexual violence and because she had that affiliation formerly with the LRA. Now, one of the things that my team and I did was ask children to draw their families during the war and after the war, because we were interested in seeing if there were any changes in family structures over time. And what was interesting about Millie's drawings was that during conflict, Millie drew her family with smiling faces. And post-conflict, she drew herself with a sad face. And this was perplexing to me because I would assume that it would be during conflict where there were bombardments, extreme violence, starvation, displacement, that during the war would be the time with the sad faces, and that during peacetime would be the time for happy faces. But it was the inverse. So I asked Millie if she could explain her drawing to me. And what she said was that during the war, she had her mother and her father. She didn't fully understand the complexity of the forced marriage, but she felt valued and loved within her family. And because her father was a high-ranking commander, she also held some degree of power and status in the armed group. This was in the contrast to the post-war period, where she talked about being a target of a stigma and violence because of her former affiliation with the LRA and because she was born of sexual violence. So Millie's drawing was vital to understanding the complexity of her circumstances and gave us the reality that for many of these children, war was actually seen as better than peace because of this stigmatization and violence that these children were experiencing. And she explained this. She said, life is hard here because people stigmatize us. They have turned their hate against us. In my family, they hate the three of us who were born in captivity. My uncle beats us and said he would kill us. He doesn't want rebel children at home. Now, Millie's story highlights that at the end of the war doesn't necessarily mean the end of violence. Now I'm going to introduce you to Sam from Rwanda. And Sam's story is going to highlight the importance of participation. So, like Millie, Sam was born of sexual violence. During Rwanda's genocide against the Tutsi in 1994, an estimated 350,000 women and girls were raped. Sam was one of an estimated 10,000 children born of these rapes. Now, in the aftermath of the genocide, many of these children were referred to and called children of hate, children of bad memories, that these children became living reminders and symbols of the suffering that survivors endured at the hands of their perpetrators. Now, Sam's mom wasn't able to explain to Sam his birth origins or what she had experienced during the genocide. It was too painful for her. So Sam explained how he learned that he was born of rape. He said, one day when I was with the other children who are neighbors, one child called me into Rahamwe. What I knew was that the Interahamwe were killers during the genocide against the Tutsi. So I went home and told my mother about what happened to me. Instead of talking, she cried a lot. The part that hurts me the most is being called the son of a killer. Now, Sam was born after the genocide, but as we can see, his daily life was shaped by the genocide against the Tutsi. And so, as we were conducting research on children born of the genocide, I thought it was really important to bring these young people onto the research team because they had so much expertise and lived experience. So I trained Sam, and my team trained Sam, and young people like Sam in research and research techniques, in ethics, in data collection. And over an extended period of time, he eventually began to, for example, collect data in the sense that he facilitated focus groups with other young people who had also been born of the genocide. And he talked about what it meant for him to be part of the research team. He said, this project was very important to me because we talked, me and other youth, with whom we share the same history. We discussed, enjoyed, we shared life challenges, and this increased the trust among us. So for me to be a researcher in this project was very important. I think this project came as a solution to us. It made the youth create a family. So in a context where this group of young people were experiencing severe isolation, marginalization, violence, and stigma because of their being born of rape, the focus groups themselves became places of belonging and solidarity for the youth. And Sam's story also brings hope and promise because after extensive research training, he was able to start a business, he attended university, and with his earnings as a youth researcher, he was able to support his family. So Sam was able to fundamentally alter his own trajectory. Now, these are the stories of 2 young people affected by armed conflict. What I want to emphasize is that there are 473 million children around the world in situations of armed conflict, in devastating situations of violence and deprivation. That means that there are 473 million stories that deserve to be told, listened to, and have ensuing action. Now, the blank canvas behind me and the one on the screen represents the future of children in war. And the question is, what is going to be on this blank canvas? Is it going to be yet more images and more artwork of young people who have been victims, participants, and witnesses to extreme forms of violence? Or is it possible that they could instead represent possibilities of renewal, rebuilding, starting anew, and a potential for remembrance, empathy, and action? It's my hope that these stories, that you can see that these stories matter, and that they foster engagement and action, and inform policymakers and decision makers to ultimately move beyond outrage. Thank you.
Please welcome Melanie Smallman, Associate Professor in Science and Technology Studies at University College London and Chair of the Falling Walls Engage Jury.
Now I can get up. Falling Walls Engage is the competition to find the best science engagement project in the world. And that's always important, to find a project that's breaking down the walls between science and society. And this year our focus was on climate change, which I think makes it even more important than ever before. So we had 15 projects from 15 different countries, which ranged from creating computer games to learn about climate change, mapping giant coral, which I learned is, I don't know if I'm going to get this right, the biggest animal on the planet. And you can see giant coral from space, apparently, so that's amazing. And we sang the Misinformation Blues. So we had a good time, despite the serious nature of the topic we were dealing with. And as usual, it was a really hard decision to find a winner, but we agreed that there was one project that captured the spirit of this year's Falling Walls Engage Breakthrough of the Year, Nondas Ferreira da Silva.
Yes, thank you. So, welcome to Breaking the Wall of Restoration with Indigenous Knowledge. My name is Nondas. I'm from Brazil. I'm a forest engineer and also founder of the Institute New Era. It's a non-profit organization that supports indigenous and traditional communities to restore nature, but also to restore hope. Now, imagine a land bigger than France, UK, and Germany together. This is the Cerrado, the savanna of Brazil, my home. Cerrado is one of the 36 global biodiversity hotspots and also home of 5% of all species on the earth. However, half of it's already being destroyed. And it's very important because it regulates South America rainfall. So we have a huge biodiversity that it's being lost by forest fire, deforestation and illegal logging, monoculture, mining, and also soya expansion, bringing my people to face thirsty and migration. And the people who have been protecting these areas for centuries, indigenous and traditional community, are often excluded from development and income opportunity. So I ask, imagine if restoration could also restore dignity and income generation. So our technique uses ancestral knowledge with modern science. We use a methodology called Muvuka. So please repeat with me, Muvuka. Yes, let's do it again. Muvuka. Great. Muvuka, it's an indigenous word that means mix of seeds. Seeds that we sow directly in the soil. It's combined this ancestral knowledge with scientific knowledge. And it's not just restoring land, it's also empowering people and engaging them in restoration activities. Until now, we have been training more than 400 people to become seed collectors through 48 capacity building training. Well, our methodology and our technique have been recognized as a good practice from Food and Agriculture Organization, the FAO. And until now, we have been restoring 50 hectares, and also training lots of people. We also have some partnership with German organization and university. And our methodology and our initiative, it's also in the framework for ecosystem restoration monitoring as a good practice. Well, here it's me with a jatoba tree. It was a seed like that I planted, and now, in 1 year, it's a tree bigger than me. Yeah, so please play the video. Good. So this is a video about the capacity building that we are doing in the rural communities. And it's not just about training, it's also about learning and exchanging knowledge with the people who are in the ground. It's also bring happiness to those people. I would like to have my maraca here. And there is a lot of knowledge about that, empowerment and also women's empowerment. And this is very important for us to create a better future, more inclusive. So, what is your goal for the future? We would like to scale up our model for 50,000 hectares, creating more than 2,000 green jobs and doubling women's participation. And when I see young people collecting seeds with pride, when I see indigenous women leading restoration, I realize we are not just restoring nature, we are restoring belongings, identity, and hope. I dream in a future that restoring nature is also restoring ourselves, because restoring nature is creating a future where dreams can come true. And everyone here can be part of that, because we are the generation of restoration. Obrigado. Hey, Klaus. You're going to give me a hug, too? Thank you.
Breaking the wall of future cities. How technology shapes the urban landscape of tomorrow. Carlo Ratti, Massachusetts Institute of Technology.
On November 9th, 1989, I was in my teenage room, watching a wall become a bridge.
All right. Good afternoon, everybody. Great pleasure to be back in beautiful Berlin. What I want to share with you is some of the work, some of the research we're doing at MIT at Sensible City Lab. Also with cities around the world. We found that in order to do research that then can be turned into action, it's really important to be close to cities and close to citizens. Why are we passionate about cities? If you need to remember 4 numbers about cities, just remember those. 2, 55, 75, and 80. Cities are only 2% of the surface of the planet, but are 55% of the population, 75% of energy consumption, 80% of CO2 emissions. So if we can do something to make our cities a little bit more sustainable, that can be a big deal. And today there's an interesting situation. The situation is that digital and physical are converging in cities. we get a lot of data, and the data can help us to better understand, to change the way we understand, design, and live in cities. So, for instance, we can look at cities like this, like living organisms. And then, that changes the way we understand not only the physical city, but the life that unfolds inside it. And we've been doing that at MIT for some time. We've been discovering a few things. For instance, this is a very interesting book by a great mathematician, Zip. And in this book, Zip discovered, among other things, that if you got two cities, the flows between two cities are similar to the gravity law. So if you take Berlin and if you take Hamburg, then the population flows, for instance, between Berlin and Hamburg can be described as population 1, population 2 divided by distance squared. Now, at the time, that's what you could do, like 100 years ago, but today with new data, you can look at new dimensions of this. So we discovered that also there's another component there that's related not only to distance, but to frequency, and how many times you go. So the invariant, if you want, is RF, not only R, the distance between the two cities. Or something else is, we could finally figure out recently, is something that bothered me since I was a student there. That's Cambridge, England, and that is Darwin College. And I realized something puzzling, that I was going from my college, Darwin College, to the department, following one route, but then going from the department to the college, following a different one. And why was that puzzling? Because the shortest path, as we all know, is symmetrical. A to B equal B to A. So you could say it was consistently inconsistent. And for somebody studying and doing rational studies, that was really puzzling. Well, again, with the data we have now in cities, we started looking at a lot of anonymized traces in Boston and in San Francisco. And we realized that actually our brain works in a way that's very similar to many animals and does not shortest path, but vector-based navigation. Vector-based means that you are looking at a vector at the objective and minimize angular deviation as you're moving towards it. And if you look at that, that is close to shortest path, as you see here, but not exactly the shortest path. And you can imagine that like tens of thousands of years ago in the savannah or in the Cerrado in Brazil, our ancestors were trying to get to the destination as best as you can. The vector-based navigation is super efficient from a computational point of view, but save again some computational power for other things, for looking around, for looking at problems, and so on. So somehow it's an interesting evolutionary trade-off between being perfectly optimal with shortest path and also keeping some computational power for something else. Those are the things we've been doing at MIT. And I will not tell you more about some of those projects. I want to tell you tonight about something we started looking at recently. And that has to do with how we look at the city. Looking at the city has been an activity that has been happening for a long time. This person, who knows who this person is? Camillo Sitte. Camillo Sitte at the end of the 19th century, 1889, he wrote a beautiful book that's still studied by every architecture student called the Städtebau. And what he did was actually go around many cities, especially in Europe, analyze them, look at the configuration, and really, at the time, look at the visible city and how it affects what we like, what we don't like, and how we behave inside it. 100 years later, less than 100 years later, in the 1960s, this other guy called Kevin Lynch, who was at MIT, tried to develop that further. An incredible book called The Image of the City. And again, how the city looks says a lot about how we understand it and how we move inside it. What he says at the beginning is this book is about the look of cities and whether this look is of any importance and whether it can be changed. And then, he looked at different things, 5 main parameters. You see them here. Here you see different resolutions of the city. And so what Kevin Lynch was looking at was, for instance, about what you see here, about paths, about nodes, about districts, about edges, and also about landmarks. So this is just to say that there is a long history of looking at cities and trying to make sense of them. And then we come to this guy in the 1970s or 80s called William White. And William White, in this beautiful book, summarized some of his findings. What he did for the first time was actually looking at video from cities. he went into New York and he started taking video. It was the very beginning, so those were Super 8 cameras. And then he looked at all the video and then started analyzing. Look at this, by the way, William White, if you've been in New York and enjoying the beautiful Bryant Park, say thank you, William White, because he helped preserve it and save it for today. And he took all this video. This is Bryant Park, you know, how people move in space. He looked at what facilitates connections, interactions, new friendships, people you don't like. He also discovered some beautiful, simple things like, you know, what attracts people, it appears, he says, is other people. And also how things change in the course of the day when it's hotter, warmer, and so on. And then he was actually looking at this and annotating all this and actually turning that into quantifiable thing. Here he counts all the people and how they're moving. You see it here, for instance, if you take the camera. That's again New York. He was looking at a lot of squares and sidewalks. And then from this, you see at the moment you can go into counting and looking at how everybody's using space. Now, when he does it in the late 70s, early 80s, he's saying in one of the notes, you know, this is taking video is fun, it's enjoyable, but actually the difficult part is how to analyze it. Evaluation is the problem, he says. Well, today, we can actually do evaluation very fast, using deep learning, CNNs, you know, classify something to understand and recognize things. This is what can be done since a few years in a very precise way. You classify different things on images, on video, and then you can also trace them in space. And so we decided, we asked a question a few months ago, which was, how has public space changed? And so we looked at the footage of where William White used to take footage, like 50 years ago. We looked at more recent footage, taken 10 years ago by a colleague, a sociologist, and we started comparing the two. And we discovered something that we didn't expect. That's a paper, if anybody's interested in looking at it, it came out just very recently. And then if you look at lingering, between 1980s and 2010, we don't linger anymore, or much less, in public space. I think because we got phones and so on, you know, it looks like we spend much time. We define here lingering people moving at less than 0.5 meters per second. So when you're going there and enjoying the space. Walking speed is faster. We walk 15% faster today. And you see it here, by the way, here you see all the different places that he analyzed, and you see to the right all the analysis of all of this. And solo walkers are more or less the same. People who are solo in public space, 67% 50 years ago, 68% today, but with a big difference that we then don't meet. If you look at that, you know, those are people, solo people who then meet somebody else, and that's a percentage, and that's how many seconds before you enter a certain space. And you see that actually public space is not anymore the place that brings us together. So this is just an initial piece of research, but then we're asking, is something fundamental changing in the way we use public space? And you know, public space is so important. Cities didn't exist once upon a time. Cities emerged around 10,000 years ago when our ancestors found a beautiful way to bring us together so that together we can exchange goods, we can exchange ideas, like today. We can exchange chromosomes. So a beautiful way to make sure that together we are more than each of us individually. And the key thing of the city, the glue of the city, since the beginning has been public space, the Agora in Athens, the place where we go. And I would say that public space is especially important today because in digital space, it's so easy. You can cancel all the people you don't like, all the people who think in a different way from you. But actually, public space has an incredible dimension that's inevitability. You need to face diversity. You meet people with different political ideas, with different people you normally are not part of your inner circle online. And so I think today is kind of an antidote against the polarization we are seeing everywhere. And so this question is what we want now to explore more. It's just the beginning. We think with AI we can start to look at some of those dimensions that people were looking at, like William White 50 years ago, and see how really this glue of the city, this is a paper not a paper, just an op-ed I did in The Guardian, if you're interested, but basically how this can help us to see in the future how public space can remain what it is, the place of encounter, and the place where together we are more than each of us individually. Thank you.
Breaking the wall of our sun's mysteries. How connecting solar observations to space probes reveals new insights. Miho Janvier, European Space Agency.
On November the 9th, 1989, I was living in Helsinki. No matter the walls, physical or mental, that we erect between us.
Good evening. It's been mostly cloudy today in Berlin, with a cool breeze and a high around 7 degrees Celsius. So keep that jacket handy if you're heading out. Now, turning our eyes beyond the clouds, space weather is looking fairly calm after a few days of disruption. The sun has been showing a few bright active regions over the past few days. Solar wind speeds were quite strong on Friday, with chances of auroras. But today, the skies above Earth seem relatively quiet, with no disruptions expected for satellites or GPS. And for our aurora fans, a small coronal hole could bring a touch of northern lights to higher latitudes tomorrow night. So keep looking up. The show might just be in the sky.
When you think about weather, you probably think about rain and clouds. Maybe images of hurricanes and storms come into your mind. But did you know that space also has its own weather? Space weather corresponds to the changes in the space environment, such as in the solar system, due to the sun's variability. This is a real footage of the sun at the center and the surroundings of the sun. And each of the puffs that you see are actual solar storms. These are happening above our heads in the sky. Now you may think this is way too above our heads in the sky. It doesn't really matter what happens in there. What I want to know, and what my grandparents always ask me, is where's the sun when it's raining here on Earth? But it does matter to you, and it does matter to me. Because space weather has a lot of impacts, especially on our technologies. It can impact satellite communication, GPS. It can also impact civil aviation, astronauts, and also electricity grids here on Earth. It's not only bad things, of course. We also have absolutely beautiful celestial displays in the shape of northern and southern lights. If you ever get the chance to see some, I highly recommend to do that. But let's go back to a real event. This is the Iberian Peninsula back in April of this year. A blackout, electricity blackout everywhere in Portugal and Spain. Okay, that is not sun-related, but this is just an example that I want to show you, because for an event that lasted less than 24 hours, the estimated economical cost of this electricity blackout is 1.3 billion euros. Imagine this at a much global scale, over a lot of different countries covering the globe. This is such an event, of course, that governments around the world have started preparing in the form, for example, of space weather prediction centers. In the word of a US administrator, a former US administrator, the keywords are to predict and to prepare. Of course, I'm not going to talk so much about space weather, because what I want to talk to you about is what we have discovered about the sun that helps us with these predictions. I am a solar and space physicist, which means that my keen interest is understanding first and foremost our star. So back to basics, I'm going to start with the light that comes from the sun. The light that comes from the sun, like any of the stars of the universe, is very varied. But we, as humans, with our own human eyes, we only perceive a small section of this light that comes from stars, what we call the visible light. This is the sun in visible light. It's probably similar to the drawings you used to do when you were little, this yellow orangey ball, where we have a smooth surface, sometimes with a few spots, what we call sunspots. There's not much happening. But remember, this is only one part of the full story. Stars and the sun emit other types of light, what we call X-rays, or UVs, or infrared, even radio waves, are part of the light that comes from stars. And this is our star, the sun, in ultraviolet light, a complete different landscape to what we have seen before. Now, not only do we see bright regions, we also see dimmer regions, and this is constantly evolving over time. I'm going to make an analogy here, and I'm going to present our planet Earth and then the sun on the side. Both are globes and both have poles. We can define the regions at the poles. And both have regions that are much quieter. A bit like on Earth, you may have regions of the globe where you don't have a lot of natural events, maybe not so many earthquakes or volcanoes. In terms of weather, it's not really the place where you would find hurricanes. And then you also have places on Earth where you have regions that are active in terms of natural disasters, earthquakes, volcanoes, hurricanes. And the same happens for the sun. But it doesn't happen, like on Earth, in the sense that these regions are constantly changing over time. I'm talking days, even minutes. Things are changing and very variable in the sun. I'm going to stop talking just for maybe less than a minute to let you appreciate all the footage that we now have of the sun, and that will hopefully let you a taste of what it means when I say that the sun atmosphere is very variable. So please enjoy. So we have some music, but it's okay if we don't. All the things that you see here in the sun's atmosphere are evolving at a scale of a few minutes to a few hours. And a key element of these dynamics is actually the sun's magnetic field. The sun's magnetic field is omnipresent. It is absolutely everywhere, and it will shape the sun's material. And this magnetic field, again, is evolving over quite a few years. Not as long as the Earth, I'm talking around 10 to 13 years, in what we call the solar cycle. In this image, you have the sun in extreme ultraviolet light, the orangey bit that is quite florid, and the one on the right side is the visible light. And you can see that over time, you've got the years. Over time, we've got more and more sunspots appearing on the surface of the sun. But what is interesting is that all these active regions, the bright regions of the sun, are also evolving over time. Again, over the scale of 10 to 13 years. And this will lead to explosions or solar storms as time goes by. So we've got a nice video here where we're zoomed on the sun at the beginning, and we see this seed of a solar storm. And then as time goes by, you see this storm that is expanding and becoming much bigger as time goes by. This is what is a solar storm. Literally some solar material from the sun that is escaping the sun and growing until it reaches, if you have a planet on its trajectory, planets of the solar system. And that brings us to a lot of questions, right? I talked about the magnetic field. How is the magnetic field generated? Can we predict active regions? Can we predict when these active regions are going to explode over time? To answer these questions, 5 years ago, we launched in space a spacecraft called Solar Orbiter, an international collaboration between the European Space Agency and NASA. I like to call it the Swiss Army knife of our community because it transports on board a lot of telescopes that are looking at the sun and also sensors that are monitoring the space environment of the spacecraft. It's literally looking at the sources. For example, if you were on a boat looking at the water falling in the Niagara Falls and scooping the water once it reaches your boat. We can do that. We are ingenious to do that. And it has really changed my life. This is a picture of me in front of the spacecraft with this cover that we call the lab coats in order to be in a clean environment. And that was just a few weeks before it was sent actually from Germany, where the last tests were done, all the way to Cape Canaveral in Florida. February 2020. We gathered family, colleagues, friends. This is an hour before launch. You're going to see two videos, one where we see the rockets at the center, and this is the view that we saw us on the benches. I think the sound is not going to work again, but you have the countdown. Maybe I can talk to you that we were completely white-faced, holding hands. Some of us were screaming, hooray! Is this normal? I hope there's no explosion. Actually, we had an extremely beautiful launch. Our spacecraft was put in a really nice trajectory, and for the next 5 years, I got to work on the mission. Now, mind you, that was in February 2020. We were very lucky, because a few weeks later, we were all locked down in our house. This is the trajectory of, so just a visual impression of the orbit of Solar Orbiter in white. And what I want to show you here is that the orbit of Solar Orbiter, as time goes by, is tilted, which means that we're getting out of the planes of the orbit of Earth. So we have now a new view, and a new view that humanity has never had before of the poles of the sun. Remember I talked about the poles of Earth? We're now looking at the poles of the sun. So what have we learned with Solar Orbiter? Many things. Unfortunately, I don't have the time to go through everything. But the fact that we're getting closer to the sun with our telescopes means that we can see in many more details the atmosphere of the sun. This is the sun, the full sun, and you can see the amount of details that we see now. This is a video. And maybe one thing that I can hint is that those tiny little brightenings that you see, we think are mini explosions, similar to the big explosions that are happening. And maybe they hold the key of why the sun's atmosphere is so extremely hot. Because Solar Orbiter sometimes goes behind the sun, as seen from Earth, we can also follow active regions that we cannot see from Earth. And that's the case for the active region that gave us really nice auroral activity last year. So you can see it with the white circle. We're following it from Earth, and then it passes behind, Solar Orbiter catches it, and it gives us, again, a missing puzzle of the evolution of these active regions, allowing us to constrain our modeling. I searched and I found that indeed in Berlin, you also had the chance to look at the Northern Lights. So who here, by the raise of hands, saw any of these auroral activity last year? There was one in May and one in October. So I see one hand, 3 hands. Not that many. Next time, put an app on your phone. We have now auroral alerts that can tell you, but generally they happen at 2 a.m. when everyone is deep asleep. And they send you an announcement saying, like, go outside and watch some auroras. We have also seen the poles, as I said. So this used to be the grazing view that we had of the poles, where there was a hint of quite some activity going on. Now we have this view. So this is a view from another spacecraft that we have. But just to show you the difference, this is the view from us, or the view from the original view that we had. And this is going to change to the Solar Orbiter view, where now we're looking at the poles of the sun in a high amount of details. And what it is showing us is that the poles are also the regions where we find strong magnetic fields, a magnetic field that is changing over time, and that may hold the key of the solar cycle. So watch this space. I want to finish with 2 beautiful images. One is an artist view of the Earth and the sun. And the message that I want you to keep is that we live in the neighborhood of an active star. Good. Don't look at the sun with your directly, even with your sunglasses. And the last one, yeah, actually, that's good. And the last one is this beautiful image that we get from the James Webb Space Telescope. That's really good, actually. But it's night time now. So now we can turn to astronomy. And this image actually shows that all these bright points, or stars, where we find exoplanets orbiting around them. And so a question for you is, are you going to be with me on this journey to understand the sun's mysteries, and also the stars' mysteries, and whether there's life on the planets orbiting those active stars out there? Thank you so much.
Breaking the wall of genetic barriers. How mRNA technology revolutionizes medicine. Katalin Karikó, University of Szeged.
On November the 9th, 1989, I was in our apartment in the suburbs of Philadelphia.
Hello. I did not come empty-handed. I get the Nobel Prize just before I forget to show, especially to the young one, before they will receive their own one. This is how it will look. I grew up in Kisújszálás, a small agricultural town in Hungary. My parents had only elementary school education. My father was butcher, my mother bookkeeper, and early on I learned that hard work part of life and how to make sausage. My parents, with my older sister and me, we lived in a small house and with big garden. And so the wonder is, how did I get from this simple life, single room house, no running water, here in front of you, telling you about my story? I was a curious girl. I liked to watch nature. I watched the neighbor cow gave birth, and I wanted to understand everything about it. So early on, when I was in elementary school, you know, my sister was very good, and she's 3 years older, and everybody, when you have an older siblings, expect that you are doing well. And I started to compete in geography, biology, and chemistry. And when I was not doing well, I always, you know, tried to inspire myself to learn more. And eventually, at 14 years old, I was third best in Hungary in biology. The competition was in Budapest, and my father put me on the train, 150 kilometers from Budapest, that go to the competition. Yes, and recently I found a book that I started to write already when I was 13. I wanted to know all about the animals, and okay, don't look very close to this lobster, it has extra legs, but that's okay, you know, you can expect this from a 13 years old. I don't even know why I started to draw animals, because I always love plants. I wanted to know the name of all of these plants, and then I had this Hungarian dictionary, look it up, and I noticed that there is an extra name, some Latin name, and after that there is a capital L. I learned that it stands for Linné. So when the high school, I was still in elementary school, high school advertised to write an essay about your favorite scientist, I wrote about Linné. I figured out how smart he is, that each living organism has to be unequivocally defined by a name. And I was just amazed, and maybe that's why I am very, very well organized. So again, you know, I already wrote a book and an essay, and so it seemed that I always set up a goal. That's like that, you know, set up a goal. I didn't get extra point for that, but, you know, set up and then submit, work on it, submit, and that's it. I never get anything from this essay on Linné. But it's not important. Important goal and work towards it. Of course, when my goals, which I set up later on in life, it was more challenging, then reality hit. And so I had to come out, you know, and break down a lot of walls. I wouldn't sit here in front of you if high school I wouldn't learn how to handle stress. And you know, sometimes just reading one book can change the life, and it changed my life forever. So in high school, we sent letter to Hungarian scientists because we couldn't speak English. And then we get response, and we were so inspired by Seje. Seje was a Hungarian scientist in Canada, and he coined the word stress in 1930. Prior to that, it was only in physics. And he demonstrated that stress can kill you. But not the stress itself, but how you perceive it. He also mentioned that you need positive stress, excitement, anticipation that we are gathering together. You know, that's what makes you get out from bed in the morning. If you don't have positive stress, then, you know, you're still in bed. And so what is important is you have to learn how the negative stress you turn to positive. And so later I learned that what philosophy he was promoting, actually it is the Stoic philosophy. You focus on things, what you can do, what you can change. And do not complain, do not blame others, and do not compare yourself. That's especially the young one I am saying. Constantly you are comparing yourself to somebody else. And you conclude that you are better, but somehow this other is advancing. If you already pay attention on this one, you did not pay attention what you can change, because you cannot change that. So I also learned, you know, that do not do revenge, slashing tire of the boss or something. Don't do that, because it hits you back. It escalates. So you have to think what Shaya suggested, to feel gratitude towards somebody. You were just ready to set a revenge, because then you can calm down and it won't hit back. It also said that don't carry grudge against anybody, because it poisons you. Another person wouldn't even remember what you are upset. So these are the things, and then one major message, I put it up there. If you want to do something, you find a way. If not, you find excuses. So just remember that when you find excuses. And so I, after, learn all this elementary and high school, and went to University of Szeged, where I started to work with lipids. I'd never wanted to work with lipids. I love plants, but, so I knew something because my father was butcher. I knew about lipids, but that was a different thing. We wrapped up DNA in it, delivered to the cells, and one day, the boss from the RNA team came and suggested that if I want to make my PhD, then I can join his team. So I am not a visionary. If end of this talk is not convinced that I am one of you, I had no special talent, no vision. I just always went with the flow. It is just, I could focus. And this is very difficult in these days because, when I was young, it was easy. There were no telephone, no TikTok and other things. But it is also very important to have a good family background. I married 45 years ago. We just celebrated this with my husband. My daughter yesterday was 43 years old. And my daughter happened to be 2 times Olympic champion and 5-time world champion. So, when they told me, a couple of years ago that, oh, Kati, nobody knew about you, I said all of the rower knew I was Susan's mom. And of course, when now she's coming with me, is usually, she's Kati's daughter. Yes. So I worked in a laboratory in the United States, different places, but in this, if you have never seen a lab, this is how it looks. It's very nice and tidy because I like order. I just mentioned Linné. And, the budget was low. You can see no lab coat, no gloves, no nothing. But I managed it. And so I worked by myself because I couldn't get grant. So I tried to explain why I didn't get grant. So because what you do your research. And you get the result, and then, you publish it. Because you have something to say and want to share the result. And then you write a grant because you want to expand your team and do more work. So from this one, I never get the big bag of money. I went back again to do more research, so it was a short circle for me. But other people get money, funding. And what happened is that as they get bigger team, now students joined, and now that they publish because the student had to graduate. And also they publish, they have nothing to say, or not much, because the PI want to promotion. And so that's how I see that, the scientific knowledge versus the career is the same path. So I try to explain why I never get that. Anyway, what happened is I think that the goal of research should be to understand nature. But the 21st century scientists want to publish in nature. So that's... Because, if your goal is to understand nature. You even happy when you see somebody is publishing what you are just working, because you want to understand, and you are happy that, oh, we have more knowledge. But nobody is nodding that, I am very happy when somebody is just scooping my research. Yeah, so but I was never disappointed because, I want to understand nature. And so with Drew Weissman, I met him at the Xerox machine. Probably the young one don't even know what this copy machine is then. But it was 1997 and only 2002 when I started to collect the digital papers and reading there. But luckily, the progress was not that fast. So 97, we still copied. And I met Andrew, was interested in developing vaccines. I want to develop mRNA for therapy. And by that time, 10 years, I worked on RNA. I did not know that RNA I am making is inflammatory. But then, we together, Drew could make these special immune cells and dendritic cells, and we found that the RNA I'm making inflammatory. And we asked the question, is all of the RNA is inflammatory? And because 10 years I already collected different RNA, so we tested out, and we found that no, not all of the RNA, the tRNA, transfer RNA was not inflammatory, and that gave the idea that maybe modification of the nucleoside is important. So we didn't set out to make a vaccine and to make it something, non-inflammatory. We didn't even know that it exists because we are scientists. We are just doing things which we believe maybe exist and trying because, we are super optimistic. Anyway, so that we changed one nucleotide in this RNA because it has 4 building blocks, and when it was pseudouridine, it was non-inflammatory. So this is what happened exactly like 5 years ago. Actually, today is November 9. If you remember 5 years ago, November 9, when it was announced that the vaccine was effective. Yes, and And here we are with Drew Weissman, with camera, and they gave us in December 2020 the vaccine, and after that, was this, a couple of years later, this Nobel Prize ceremony, and whoever is important. My husband, my daughter, who are always supporting me, my sister was there. Those are in the middle. Is it my teachers from University of Szeged, and those who supported me in University of Pennsylvania. My daughter's in the middle, again, on the ceremony, she overshined me. She got the best dress, voted based on the TV viewers. Yeah, so that's my life. And recently, and actually, last month, she got a statue in Hungary, for the achievement I didn't get yet. Anyway, so this was my life, and I specifically made this for you, timeline of my career. At the beginning was nice and up. At age 30, they dismissed me from Hungary, and I lost my job. And then I found another job in the United States. And then 3 years later, my supervisor, he loved me very much because I worked day and night, and he wanted to keep me. But then, when he learned that I get the Johns Hopkins offer, he made sure that they will withdraw that, and I was subject deportation, and he kicked me out. And then I found a job in University of Pennsylvania. So this is what happened. They kicked me out in Hungary. Finally, I get faculty in the Ivy League school. And so there, again, the happiness was not too long because, they demoted me from the faculty position. But I said, the bench is here. I am in the United States of America. Where else, if not here, I can do the research? And so I did major discoveries. And then at age 58, I was kicked out. I was forced to retire because I was not getting the money. You know, the bag was missing. And then, you know, I tried to go somewhere, and because I felt that I still have a lot to offer, and that's how I ended up in Germany. You know, left my family behind, and in an attic, you know, for 3 months, every day I went to work dressing up from my suitcase. And that's, you know, prepared me for those awards things, which was recently constantly dressing. And so this is just telling you, you know, this curve, ups and downs, and so how many times I hit the bottom. And what is important is, listen, you might get kicked out. Don't feel sorry for yourself, don't complain, and don't spend any energy on try to justify, try to get it back. It is done. All your energy is what next, because that's what you can change. And that's how I did also. Thank you. You know that from all of these flowers, I have to say something. Yeah. But thank you. Once my daughter told me that we were watching the Winter Olympics, and those ice skaters were beautiful and jump up and down and everything. And my daughter said that, I want to be ice skater. I have to say she's 6'2", so she's 186 centimeters. You haven't seen it. And then I said, why? She said, did you see, Mom? They throw all of those flowers to them. And thank you. And I told her that, hey, if you like the lot of flowers, you have to be gardener. Thank you very much. Thank you. Thank you. Thank you.
Please welcome Andreas Kosmida, Managing Director of the Falling Walls Foundation.
So, my drama teacher once told me a tough act to follow is not a bad thing. Everybody will be energized. So thank you for that. That was amazing. So guys, but do you have enough energy after these 4 days for one last cheer for me? Do you have it? How did you like this summit? Come on. Woo! So, I was asked to wrap this up. Right, so let's wrap it up. As you maybe know, I'm in charge of the numbers here, so I will share some with you because they're actually quite impressive. So number one, or the first number is 2,270. 2,270 participants in this summit. That's you guys. That's amazing. 2,270 individual voices, ideas, minds that came together from 97 countries to this very breakthrough city of Berlin to make the world a better place, to make walls fall. Thank you guys. Without you, this would be an open house and nothing. You are the material that's going into this optimism factory, and you're also the product. So you feel special about that. Thank you so much, 2,270 individual people here at this Falling Walls Science Summit. Woo, crazy. All right, so we really stepped up the game, 4 days. You guys are troopers, okay? That was a marathon. We did 134 individual sessions with 774 active speakers in all these stages. Thank you so much for everybody contributing. You were amazing. The overall breadth of the program, thanks to you, was never like this before. I go out of this conference with so many ideas, with so many new inspirations. And as we said so many times, yes, there are quite a number of challenges in the world. And yes, it's very easy to become a little gloomy at times. But if you take something away from here, one call to action, one new project, one new partner, one new idea, that all made sense and it was worth all the effort. So I hope you take all these great ideas with you. I hope you do something with it later on. I'm always asked, what's the impact? So I will ask you next year what you did with all of these things that you took away from here. Thank you to 774 speakers. And of course, we need to thank quite a number of institutes, universities, entities, foundations, the government, the state government, the federal government, and many, many others contributing. As you can imagine, this does not come for free, and everybody you see on this board contributed in one way or another. So thank you. Give it up for the sponsors. Why, look at that. There's still room. Ha! So, maybe somebody's not on the board yet, and maybe somebody would like to be for next season, because it will also be amazing, and I would be the guy to approach to fill this little gap down there. Can you do that? All right. Thank you so much in advance. Right. I'd also like to acknowledge the founder of the whole thing, Sebastian, 16th edition. I see you smiling. Thank you so much for kicking this off. Round of applause for Sebastian. And also, obviously, we need to acknowledge a fantastic chairman of the board, guaranteeing the quality, bringing all the jurors, Jürgen Lünneck. And now, for maybe the most important person of this summit, can I get some flowers? I need some flowers. I actually had some other flowers, but I'll take these flowers. Oh, no. Thank you so much, but I will also take this. Right? Right, so I guess you agree with me that everything ran quite smoothly here. And that was quite an effort, right? As you can imagine. And there's one person who is conducting all of this machinery, who's sitting in the basement with the thing in her ear and orchestrating everyone around. And you need to give me a final, vast, huge cheer for the woman who made all of this possible, Luca Litzercher.
Thank you very much. I thought it's getting easier. It's my third time on the stage now, but it's not. I'm trying not to cry today. I just want to thank, on top of the people Andreas thanked already, all the people in the backstage areas, all the teams working here, our agency, technical staff, catering. We are working with around 370 people here for the whole 4 days. It was a great pleasure to work with all of you. Thank you very much for your support. And of course, the large team of Falling Walls. I really want to thank you, all of our teams. You did a great work. We all did this for 1 year, for these 4 days. Thank you very much. Please come up on stage, all the Falling Walls teams.
Give it up for the team.
There's more space in front. Come up, we take a picture.
The Falling Walls team, ladies and gentlemen.
Thank you.
One last thing. We have to say an additional thank you. And with a tear in our eye, also goodbye, because Luca, after bringing this summit to this location, reinventing it basically, and doing so much amazing work, you will leave us after 3 seasons, and we want to take the opportunity to thank you here on stage. And please join me in another round of applause. Solo for Luca.
Thank you.
And that's a wrap, ladies and gentlemen. We hope to see you all again. It's always on the same day. No excuses. I didn't know when it was. Breakthrough Day. Breakthrough Day is always November 9th. Let's see whether we keep it up with 4 days, but at least 3 days. So 7, 8, 9 is the calendar to be booked in right now. Thank you all for everything that you contributed, that you did. This is a group effort. Take this energy, these ideas back home, and we will see you again next year. Will you be there with us?